So tonight we have our 4 very special guest panelists. 4, 5. Are there 5 of us? No, there's 4 of you. Oh, we'll figure it out as we get through. My name is Megan Curzens. I'm the Executive Director of the Cumberland Community Forest Society. Normally I jump up on a stage with a Cumberland Brewing Company pint in my hand and welcome you all here. Tonight I am doing it from my office, but I set up some twinkly lights because I thought that would kind of remind us of what it would be like. So there are some twinkly lights, and if anyone else feels inspired to do strange things with their backdrop, please do. We're going to get right underway with our first presenter. As Yarrow has mentioned, we really do encourage chats and comments, and we will leave some space for Q&A after each session. And I also really want to remind people that this night doesn't end at 9 o'clock. You know, we have the amazing folks at Lucky Village Enterprises. For local folks, that's Steph and Robin, who have produced a very special show for you this evening of both music and visuals. Appropriate with all medication, apparently. That was one of the notes. So we really do encourage you to stick around or take your screen out onto the back deck or any of that. So I think what we will do is get right away into the content. One of the things that I have discovered, maybe knew a little bit going into the weekend, but feel so much stronger now, that this entire event has become a metaphor for our relationships with each other, especially in this time. This web of connection where we pop up in these various places, and it's a decentralized way that we pop up and manifest ourselves, but we are not forgetting that we're part of an interconnected whole. And that interconnected whole of community, and the Cumberland Forest Society is so about a community, but it's also Vancouver Island. And to be honest, being forced online has reminded us that this community extends beyond Vancouver Island, extends North America and beyond. And so as much as there's loss in us not being together, there's this incredible opportunity to recognize how this web of connection extends with infinite possibilities right around this planet Earth that we share. So I bring that as optimism to the night. You know, we come through this weekend, maybe not doing things the way we would normally do them, but we're reminded of these deep and profound and unbreakable connections that exist between us, and can maybe take advantage of some of the opportunities to get to know each other. I would also like to acknowledge that we are holding this event tonight on the traditional territory of the Comox First Nation, and with great gratitude we work with the Comox First Nation and respect the incredible opportunity to play, to work, to do our research, to be creatives, and all the places that they connect with us. We have immense gratitude for that, and feel very, very privileged to be on the land and the water and in the forests of this incredible Vancouver Island. So thank you, Comox. Once you start talking. Okay, Dr. Shannon Berch is a real pro and ready to jump in at any moment when asked to. She's an Emerita Scientist with the Government of British Columbia and an adjunct professor with the University, with the Government of BC and adjunct professor with UBC. She's a founding member of the Truffle Association of British Columbia and part of the research that developed that program. She's a very active member of the South Vancouver Island Mycological Society and on the search for native truffles throughout British Columbia. And it is truffles that she's going to talk about tonight. And I am going to pass things over to her. Okay, and it's over to you, Shannon.
All right, thank you. So the title of my talk is Truffle Tales. So I'm going to start out just by going over the basic concept of what a truffle is. Basically, in the kingdom fungi, many lineages over evolutionary time have given rise to truffles. And so generally they are mushrooms that have gone through modifications through evolutionary time to become truffles. And I just show you pictures of some parts of that evolutionary trail, just to give you an idea of what I mean. So up at the top here is a typical mushroom. It has a cap, it has a stem. This one has gills on the underside of the cap. And on those gills, the spores are being formed. The spores get shot off into the air. They move through the air. They land someplace and germinate and start another colony of the fungus. So this stalk that you see here, its job is to elevate the spores above the surface of the soil so that the spores can disseminate. Well, It's believed that in response perhaps to a drying environment, some fungi stopped making a fully developed stem and cap, and they started to look at like things like this next one, where you see a bit of what might have been a stalk-like thing originally, a bit of cap material. But what the place where the spores would have been formed is all convoluted, and the spores are formed inside of this mass underground, so they're not released into the air. Here's another step even further along this evolutionary line, where you can see just the remnants, just some branching remnants of what at one time might have been a stalk. And eventually the ultimate truffle is just that. It is a potato-like structure that forms underground, and the interior of it is a mass of hyphae that give rise to spores. And those spores are not shot off into the air, so they have to disseminate some other way. So we'll get into that in a minute. So just to show you what some real mushrooms, some real fungi that fit into these various categories, up at the top is everyone's favorite mushroom, the fly agaric with the cap and the stem, and underneath here would be the gills where the spores are formed. This little beauty is a species of Thaxterogaster. It's a relative of Cortinarius, and you can see that what used to be gills has been highly convoluted. This is a Gaudiaria species. It's a relative of Gomphus, pig's ears. Even less evidence of there ever having been a cap or a stem. And finally, the ultimate truffle, here represented by a Rhizopogon species. Rhizopogons are relatives of Swillus, slippery jack. So you can see there's different lineages within the fungi, all of which have given rise to these truffle things. Okay, so the truffle fungus is also a fungus that forms ectomycorrhizas with certain trees. And for those of you who are unclear about ectomycorrhiza, it's a mutually beneficial symbiosis between a particular fungus and a particular plant host. And here we see a photograph of some beautifully developed ectomycorrhizas of Tuber melanosporum. That is the Paragourd black truffle. And it's been inoculated and is forming mycorrhizas on hazelnuts. And these are some plants and the mycorrhizas produced by Dr. Charlie Lefebvre at New World Truffiers. Now, if we took one of these ectomycorrhizas and we looked at it in cross-section, this is the kind of thing you would see. Here at the center is the vascular tissue of the root. There's an endodermal layer, cortical layer. Right at the very outside is the epidermal layer. But outside of that, there is a layer of fungal tissue called the mantle. And that entirely separates that young feeder root from the soil. You would also see hyphae growing out from the mantle into the soil. And out in the soil, those hyphae take up water and nutrients and transport them back into the mycorrhiza and give those to the plant. You also see that some of the fungal hyphae have grown between epidermal cells and around cortical cells, forming a network totally covering these root cells. And this is called the Hartig net. And it's at the Hartig net that the nutrients are exchanged, sugar from the plant, water, nutrients from the fungus. All right. This is an illustration of what the truffle itself looks like. This again is a Paragourd black truffle. Here's the outer surface of that truffle, the peridium, this beautiful black, shiny, warty surface. But the interior that you can see here in a mature tuber truffle is this beautiful network of veins that look white and dark. And if we magnify that a little bit, here we can see it in this next picture. Here's the peridium, the outer surface of the truffle out here. And in here you can see the gleba, the interior of the truffle, the white veins and the darker ones. And if we magnify a couple of those a little bit more, we see that the white area is just fungal hyphae, and the darker area is where the beautiful spores of this truffle fungus develop. So the spores are in the truffle. They're not released into the air. They're underground to live out their destiny and start a new colony, they have to be moved from where they were formed to another location. So how does that happen? Well, one of the super cool things about truffles is that they have these incredible aromas. And there's been quite a bit of research on the aromas of truffles. And some recent work has shown that there can be over 200 volatile organic compounds identified in fully mature truffles. So the aroma of a truffle is very, very complex. Now, one of those compounds is a steroid, the little box, okay, called androstenol. The little box came down and blocked the word, so I lost it for a minute. Androstenol, and here's the, pardon me, the chemical structure of androstenol, and apparently it has this pronounced musk-like scent. And not only is this compound found in truffles, but apparently it's produced quite abundantly in boars and, fascinatingly enough, in the armpits of men. All right. So I'm going to take you into a little bit of science now. I'm going to talk about a couple science papers, kind of scientists who are kind of having too much fun. So this paper is about the human social attitudes being affected by, pardon me, androstenol. Hang on a minute. Can I mute myself and cough? All right, we're good to go. Thank you. All right, so these researchers in the UK published a paper in a journal called Research Communications in Psychology, Psychiatry, and Behavior in 1978. And, in this study, they took a group of men, volunteers, and a group of women, and they had them wear surgical masks. Some of the masks were impregnated with the androstenol, and some of them were not. And then they showed these test subjects what the authors describe as women in their normal clothing. And then they asked the test subjects to determine the sexual attractiveness of these women. And what they found was that those who had sniffed the androstenol gave higher grades than those that then did the others who had not sniffed the androstenol. And they speculate that this might account for the high human predilection for truffles. And in another study, a couple of years after this, a group of German researchers looked specifically at truffles. The secret of truffles, a steroidal pheromone, ? And in the introduction of the paper, they talked about the fact that in boars, this androstenol is synthesized in the testes and secreted in their saliva when they court females. And to quote the authors, the substance's musk-like scent emanating from the saliva foam is smelled by the sow and prompts her standing reflex. Enough said about that, I imagine. Anyways, what they determined in this paper that androstenol is twice as abundant in truffles as it is in the blood plasma of boars. So it's a major constituent of the aromas of truffles. And these authors speculate that this offers an explanation for the ability of pigs to detect truffles growing as deep as 1 meter underground. Okay, now it is true that traditionally in Europe, in France and Italy, pigs were in fact used to harvest truffles because pigs are very good at finding truffles using their incredible sense organs there, and they root around with their snouts in the soil. The problem with using a pig, well, there are many problems with using pigs to harvest truffles, but one of them is that pigs actually love to eat truffles. And so apparently if your pig is trying to eat a truffle and you want to stop it, there can be some very unfortunate negative consequences of this, as you see illustrated here. The other problem is if you want to transport your trained pig to somewhere else, how are you going to get it there? If you put it in your car, things could get really messy. So nowadays, most people use dogs to harvest truffles. So on the left, you see Brooke Page and her fantastic truffle dog, Dexter. And on the right is John Kelly with a superb truffle dog, Maki. These are our two best truffle dog teams in the province. Dexter is kind of the grandfather of truffle detection here in the province. Maki's a bit younger, but he's a superb truffle dog. Dexter's a puggle, so half beagle, half pug. Maki is a Lagotto Romagnolo, and that is a breed that's called the Italian truffle dog. So, you know, if you're interested in finding truffles and you want to get yourself a dog, you could go out and spend thousands of dollars on a Lagotto Romagnolo, or you could just take any dog with a good work ethic and train them, and they're perfectly good at finding truffles. So there's two kind of major categories of truffles here in BC. There's a group that are cultivated, and then there's a group that are native and wild harvested. So I'm just going to talk about those two different groups a little bit. So in the cultivated truffles, I've got illustrated here two of the truffles, Mediterranean truffles that are cultivated in BC, the Perigord black truffle and the Bianchetto truffle. There's also Burgundy truffles being produced at a truffle orchard on Vancouver Island. And down here amongst the native truffles is a Western white truffle here on the right, on the left, pardon me, and then on the right, the Western black truffle. Western black truffles are really interesting truffle because when it's mature, to me, it smells very much like pineapple. So it's quite unusual. So in the realm of truffle cultivation, the very first Perigord black truffles that were produced in a truffle orchard in BC were produced in Abbotsford at an orchard established by Bill Stewart, who you see here. The truffles were found in March of 2013. This magnificent dog that he's posing with, that's Bill's dog, never did learn how to find truffles. So a bit of a faked picture there, fake news. Here is the dog Lolo, also a Lagotto Romagnolo. Lolo is the dog who found the first truffles at Bill's orchard, who is trained and belongs to Alana McGee here. Sadly, very, very sadly, a year or two after those truffles were found, Bill's orchard, which the trees were all hazelnuts, they were decimated by a fungus called Eastern Filbert blight, which has really decimated the hazelnut industry in British Columbia. So very sadly, that orchard died off. However, not all is not lost. There are other orchards now that are starting to produce truffles in BC. Here's a great example on Vancouver Island near Nanaimo. The farm's called Below the Nut Farm. Apparently his friends and family called it the Nut Farm when he told them they were going to grow truffles, but now they're successful. So it's called Below the Nut Farm. They grow both Perigord black truffles and the Burgundy truffles. And contrary to most everybody else in the truffle world, they actually use a pig to harvest their truffles. So illustrated here are their two Vietnamese pot-bellied pigs. The white one here, that's Stevie. He's a boy. This is Annabelle, the gray one over here. They tried them both in the truffle orchard, and given my story earlier about the pheromones and the sterol, you might guess that in fact it's Annabelle who is the truffle pig. Stevie couldn't care less. All right, so moving from the cultivated truffles to the native truffles, here we see a picture of me. This is a second growth Douglas fir forest, and you see me learning to appreciate the aroma of our native black truffle, and possibly hoping that this will make my colleagues seem more attractive to me. Down here we see Brooke Fochuk and Dexter and John and Maki, both of those teams out harvesting native truffles. Now, they both tell me that it is hard work to train a dog to reliably find truffles, and even harder work to find forests where they can reliably find truffles. So they spend a lot of time out in the woods looking for good productive spots. But one of their motivations, one of the general motivations for looking for truffles, I'll illustrate here. Pardon me, on the left, we have a native Western white truffle. This one was found by Dexter and Brooke. It's 3 3/8 ounces in weight. At $85 an ounce, that particular truffle is worth about $300. So hence why some people are motivated to try to find truffles. On the left, we have a native Western black truffle found by Maki and John. It's 4 1/8 ounce. At $55 or so an ounce is worth over $200. So they are very valuable commodities. So if you happen to be a lover of canines, and you love to be out in forests, particularly Douglas fir forests, and you really like a good treasure hunt, maybe it's time for you to start truffle dog training. And there's various ways of training your dog to find truffles. It's basically just scent training with the emphasis obviously on truffles. And once you get your dog to a certain level of competence, you can enter them in a truffle dog competition. What's illustrated here is the folks and their dogs who are competing in 2018 in what is called the Joriad, which is an annual truffle dog competition held at the Oregon Truffle Festival. And if you kind of look at the people and the dogs, you see that it's a real mixed bag. You got some purebreds, you've got some mutts. This might be a Lagotto Romagnolo back here. But here's a tiny wee little guy in the front. And just to encourage you that any dog can learn how to find truffles, I will end with this picture. That little guy in the front, that's Gustav. He's at the time a 4-year-old Chihuahua mix and the 2018 Joriad champion for truffle hunting. So any dog can learn how to find truffles. Happy truffling. Thanks for your attention.
Yay! Awesome! Thank you. We can clap at home, even though we can't hear each other. I just know that everything I love is a mutant. That's what my big discovery is. Wow, thank you. Lots of thank you and very cool. Ooh, there's one. What about the Douglas fir? What is it about Douglas firs that native truffles like? That's a question from Jason Straka.
Yeah. I just mentioned a few species of truffles. We probably have hundreds of species of truffles, and honestly, we are just scratching the surface of our knowledge about truffles in the province because we don't have yet quite enough coverage of good truffle dog teams around looking for truffles. So there's lots of truffles that are not popular culinary truffles for humans. They may be great for deer and squirrels and such, but for humans, they're not all that interesting. I suspect that there's a couple things going on. One, not many people are finding truffles and trying some of these other truffle species, which may be quite delicious. But the other thing is that Douglas fir is well known to form ectomycorrhizas with a variety of different fungal species. So the ones that the current popular culinary truffles, native truffles, just happen to be associates of Douglas fir. They can also form mycorrhizas with some true fir, Abies species as well.
So Jane, do you want to come online and ask that question? We can unmute you. Do you want to ask? Here we go. Hi, Jane. Oh, we gotta wait. Hang on. Wait. Takes a second. Why aren't you in? There we go. Ahead, Jane.
So if you find one truffle, are you going to find more in that area, or do they grow singly?
Yeah, there's usually more than one. And okay, so truffle dogs, you train them to find mature truffles. So you train them so that they're only finding and indicating to you the truffles that have the really nicely developed aromas. So if you, and it used to be that for scientific purposes and sadly for commercial purposes as well, people used to use rakes and rake the forest floor looking for truffles. And if you do that, you might find a ripe truffle or two, but you will probably find lots of other truffles that are immature. And an immature truffle has no particular interest. They don't have the aromas of the mature truffles. So yeah, you can find more truffles around, but if your dog just indicates on, this one truffle, it's probably the only one of those truffles that's mature right now. So come back later, and because they take a while to mature in the ground.
Yeah, so I was just going to follow that up. How long does it take for a truffle to get to a mature stage?
Yeah. It would be lovely if we actually had good answers to that, Jane. The truffles that are best studied are the European cultivated truffles. And the Perigord black truffle is well known to start the very primordia of the truffles. They can be found in the soil in June, and they're harvested mature January and February and into March. So it takes a long time for the truffle themselves to develop, and they're really not very mature until into December, January, February. So yeah, they could be full-sized before that, but that maturation takes weeks, months.
Thank you.
All right, we've got two more questions.
Okay.
It was Gary Oaks. So the question was, what about Gary Oak groves? Is it truffles or is it something else?
Oh, yeah, a big diversity of ectomycorrhizal fungi associate with oaks, including a bunch of our native truffle species. Yeah, so we have found in a study I'm working on right now quite a few different species of tuber that are native here, but they're not big, really attractive truffles. So Gary Oak forms mycorrhizas with a big diversity of different kinds of ectomycorrhizal fungi, as ascomycetes, Physarum mycetes, mushrooms, crusts, truffles. Yes.
Awesome. Great. Lisa Levesque, you have a question. So you can ask that yourself as well. I'm going to unmute you. There we go.
Hi,
Go ahead,
Shannon.
Lisa.
I'm just wondering, are there any other areas of the province where truffles are more common than other areas? And I'm just wondering if they have any other associations, like recently burned areas or things like that, like morels or, like pines do, sorry.
Yeah, it's a well-documented fact that there are more truffles where there are truffle dogs. Joke. The honest truth, Lisa, is that we just have not explored much of the province for truffles. So, I would feel uncomfortable really to try to address that question until we get more people out there looking for truffles. I mean, we know from lots of other research where people are identifying molecularly the ectomycorrhizal fungi and the roots of spruce and pine in the interior, and they often find truffle fungi, hysterangium species, rhizopogon species, etc. So we know they're out there, but we don't have a lot of people out there collecting the fruiting bodies.
Well, that is awesome. I think on that note, I'm going to actually do one more question from Lisa, which I can unmute you, Lisa, as well, if you want to ask that. I just have to find you. Where are you, Lisa? There you go. I'm unmuting you. You have to accept it. There you go.
Okay, thanks, Megan. I just asked if the Indigenous people used truffles, and if so, what uses did they have for them?
Yeah, I don't know of any research that has shown that Indigenous peoples here in North America use truffles. Yeah, and that may be simply our ignorance.
Thank you.
And so I'm going to just ask one more on behalf of Ellen, which is, are we aware of any poisonous truffles or truffles that can make you sick?
I love that question because I've asked that question of every truffle expert that I run into around the world. So I don't want to use too many names if people aren't familiar with fungi, but there's truffles that are related to very many different lineages of fungi. So why wouldn't there be truffles related to the Amanita phalloides, the deadly poisonous truffle? We know that there are truffles that are closely related to Cortinarius, and there's many species of Cortinarius that we think are poisonous. The thing about truffles, though, is that they are co-evolved. It's a three-way co-evolution: a plant, a fungus, and an animal for the distribution of the spores. So if you're a poisonous fungus and an animal digs you up and eats you and dies before it can distribute your spores, it's just not a good thing. So I think evolution has probably selected against poisonous mushrooms forming mycorrhizas. But there's certainly plenty of poisonous mushrooms forming truffles, but there's certainly plenty of truffles that the human palate doesn't like. I mean, there's some species of Gaudiaria. Friends of mine in Oregon, truffle nerds, truffle scientists, played a joke on a friend once by hiding, Andy will laugh at this, I think, hiding a bunch of Gaudiarias under the seat of his car. And it's just the most offensive, powerful smell. So I guess maybe animals like it and dig them up, but the human palate it's not good for the humans.
Well, on that note, that was fantastic. Shannon, you're a Cumberland Forest Science Pub natural. Someone said there, Kara's fun science pranks for everyone.
Yeah, right.
Kind of stuff we're looking for. So once again, thank you so much for being part of it. And who knows, at the end, we may circle around for a few more questions and stuff, but we're going to keep flowing forward. I didn't mention at the beginning, we will, after our third presentation, we're going to take a very brief break. Not that you couldn't leave now, but in case you're like riveted, stuck to your chair, oh my God, I can't leave and refill my beverage, that we will, after Jason, take a very brief musical and visual interlude so as to refill whatever it is we're consuming. So I'm going to get right back into the next presenter. I'm very excited to introduce Dawn Copeman. Dawn is a Cumberland resident and a wonderful active member of the Cumberland Community Forest Society. We've taken to calling her our naturalist in residence. I don't know if she's accepted that moniker yet, but and she's also a photographer who's been capturing beautiful photos of the forest and really some amazing stuff related to slime molds in recent years, but all kinds of photographs. Dawn is also a community historian who does walking tours and history tours related to the Cumberland Forest, and more specifically the Chinese and Japanese settlement communities that were once here. So she just has this incredible broad-based knowledge of the community. You find her on Instagram at Cumberland Forest Hag, but she is definitely one of our protectors of the forest and embodies that. And it's a total delight to welcome her to share her knowledge about slime molds.
Well, thank you very much for the introduction, Megan. Of course, I'm completely unqualified to talk about anything scientific here because I'm strictly an observationist, and I want to set that straight right from the get-go, because if you ask me something and I'm going to pronounce something incorrectly, you'll just have to excuse me for that. Since I retired in 2014, I hike in the Cumberland Forest pretty much every day. And hiking every day, after a while, you start to see patterns and familiar plants and interesting things everywhere. So being a good little researcher, I took a few courses and bought some reference books and joined a number of naturalist Facebook sites. I talked to the other forest enthusiasts, and we share knowledge and sites of interesting stuff in the forest. Every time I walk, I find something new in the forest to be excited about, whether it's trees or plants or lichen or mushrooms or birds or animals or whatever. Megan asked me to talk about slime molds because they're my current favorite. They are great to observe in the forest because, unlike mushrooms, they are difficult to pick, so they tend to stay at least in the same general area, which is great. And in their eating stage, The worst thing that will happen when you're trying to photograph them is that they'll have a few poke marks on the surface, because people look at these things in the forest and say, what is that? All the photos in my presentation are current photos from pretty much August, September, and actually even this morning, which is prime slime season. And they're all this year with a couple of exceptions. Some of the slime mold specimens that I found in past years, I haven't found this year yet. And I have researched the scientific side of slime molds for this presentation, but as I've said, I'm not a scientist. So if I mangle the Latin, you'll just have to forgive me, get over it, and hopefully learn to enjoy slime molds as much as I do. And I'd also like to point out that something that might not be obvious to new forest users. Yes, there's a lot of interest in foraging and wildcrafting, and all this is great within limits. But the real beauty and wonder of the forest is being out in the forest without an agenda. Time stops. You breathe in the fresh air, you listen to birds and the rustle of leaves and the trickling of water in the wetlands, and yes, for me at least, I watch slime molds move. I promise you, if you've never noticed slime molds before today, you will now. You too can become a proficient slime molder. Okay, I'm gonna go next. Okay, why do I like slime molds? Now, this might be a fairly strange thing to show up on the screen, which is the blob. But one of my earliest memories is going to the show, which is what it was called then, with my older sister to see the movie The Blob. It was one of the first appearances of Steve McQueen. It wasn't a first-run theater. This came out in 1958. But my mother had 5 children really close together. So throwing a couple of quarters at the 2 oldest to go away and watch a movie for a couple of hours was probably a good idea. I was maybe 6 or 7 at the most at the time, so not very old. And I just checked about this movie online before this presentation, and it's actually rated R. But of course, it was the 60s, so nobody paid any attention to that kind of thing. My older sister hid under her coat through most of the movie. She said, tell me what's going on, tell me what's going on. But I was riveted to that screen, and I gave her the play-by-play about the blob. That must have been the spark for my interest in all things weird. I don't know. It's amoebas from outer space. Okay, I thought about that movie researching this presentation because there was a very well-received exhibition in Paris last autumn that was called La Blob, with all sorts of amazing information about slime molds. You can check it out online. Some of the facts on La Blob are eerily like that 1958 movie. Well, I'm kidding, sort of. But as you can see, it was based on a true story of a blob from outer space. And it doesn't have a happy ending. Well, it sort of does. The blob gets, I won't spoil it for you. You can look it up online. Okay, so what exactly is a slime mold? Where do they fit in the animal kingdom? Actually, the answer to that is they don't. They were originally in the fungal classification, named molds because they spore, but like poor old planet Pluto, with its demotion from a planet, slime molds were kicked out of their fungal classification and oozed into the kingdom of convenience, where all odd things end up called protista, which is a group of organisms that are unicellular or unicellular colonial and form no tissues. It's a kind of none of the above category classification, as other members don't share a common ancestor. It's basically where the weird things are. I got a quote from Fred Spiegel, who's a professor of biology, retired at the University of Arkansas, from the slime mold Facebook page. The term slime mold refers to any organism that lives its life as an amoeba and then makes a dispersal structure that looks superficially like a fungus. The slime part of the life cycle is the amoeboid part, the slimy part, and the mold part is the spore-bearing dispersal structure or fruiting body. So I hope that's clear so far. What I want to show you is a group of slime molds, which are currently a count of around 100 types, I think, called the Myxomycetes, Myxos for short. They live most of their life as microscopic, single-cell, free-living amoeba, but then something magic happens. They mate and form structures that are still technically individual cells, but they change and transform into a large cell clear mucus, one giant slimy amoeba called the plasmodium, which you can see in this picture. Okay, Physarum Polycephalum translates as a many-headed slime. It refers to the behavior of Plasmodium. They are amazing creatures. They move to and hunt down and ingest food in the form of yeast, bacteria, dead plants, and fungi. That's right, they move. And quickly enough to be seen with the naked eye. They forage by branching out in fractal patterns, using their undulating little arms to find and surround any edibles. Slime molds move around by pulsating, circulating water through its one enormous cell to push some kind of signaling molecule, it's probably calcium, back and forth through the organism. The calcium triggers contractions to the outer cell walls and starts a cycle of pulsing and squeezing, referred to as shuttle streaming. I get an image of a boa constrictor when I think of this movement, but one with many heads, like a hydra, but cuter. When faced with an obstacle or a forked path, slime molds, unlike humans trying to solve a labyrinth, don't have to choose one way or the other. They can branch and take all paths simultaneously. If obstructed, they branch again. After diverting themselves around an obstacle, they find the shortest path to the food. It's like the slime mold sends out food scouts in exploratory mode in all directions, pruning back on less successful forays and reinforcing the networks to the shortest paths to food. Once the food is found inside decaying logs, the slime mold grows over them and begins to digest them through its body, just like the movie The Blob. Amazing. It is the ability of slime molds to remember, decide, and solve complex optimization problems, such as transit planning, that has researchers really excited. Slime molds can make decisions by comparing a range of possible courses of action and finding the shortest path between 2 points. They leave chemical trails to mark dead ends too. This behavior can be replicated in the lab on agar plates using yummy oat flakes as food, and slime molds all love Quaker oats. Researchers in Japan placed food scraps deliberately to replicate the locations of some of the most visited sites in and around Tokyo. Any obstacles, such as mountains or other natural obstructions, were taken into consideration, and they used light or salt, which slime molds don't like. They stood in the places where there were natural obstacles. The plates were inoculated with slime mold at the point representing Tokyo, and in the first few hours, the slime mold did its fractal seek food trick, and tendrils branched out through the entire map. Within 26 hours, the size of its branches started to shrink, and the slime mold created a complex branching network that was reinforced along the shortest pathways to the food. So the mold recreated an interconnected network made of slime that looked almost exactly like the efficient, well-designed Tokyo subway and rail system. And they did different trials, and the networks weren't always exactly the same, but the networks designed by the slime molds were just as efficient. But of course, it gets weirder. Not only are slime molds great at solving labyrinths and designing efficient transportation systems, they can learn. In one experiment, researchers kept placing slime molds next to food blocked by bitter but harmless substances such as caffeine. After a few days, the slime molds figured out they could shuttle across the streams of caffeine, no problem. It was worth the effort as long as the food was high value. Slime molds also don't like salt, but with high value food on the other side of a salt bridge in another experiment, they developed a resistance to the salt over several days. And they remember new information, this information trick, even after several days without practicing, and even after a hibernation period where the slime mold is dried up and inactive. The original slime mold can teach other chunks of slime mold too. The separately inoculated slime mold also has this knowledge about tolerating salt or caffeine. It's still not clear how this works without anything we recognize as a nervous system. So no brain, no problem. The human brain has 86 million neurons, and we've always considered ourselves the top of the heap on this planet when it comes to intelligence, right? One slime mold researcher who was constantly getting lost in IKEA decided to train a slime mold to find the exit for him by creating a maze of the floor plan. Yes, the slime mold figured out what the researcher could not. And there's this place that I found about, there's a place called the Unconventional Computing Laboratory at the University of the West of England, which I just want to go there, the Unconventional Computing Laboratory. Scientists there have used slime molds to calculate efficient fire evacuation routes from buildings, and are also working on organic computers, solving complex mathematics and programming robots, making decision gates and oscillators, and also modeling possible patterns of human migration, whether through climate change, designs of supply chain networks, or planning the most efficient paths to the moon. They also act as sensors on computers, so there's network-based organisms that they're using for computers. It's just so many possibilities. Now, to be fair, mycelium is better for long-term studies because the pathways don't morph and change and adapt quite so quickly. But slime molds are the torchbearers and used in cutting-edge research. And we'll go to slide 5. Okay, so that's quite a bit of chewy science. We've got something a little bit lighter spirited here. Okay, this slide shows the progression of most of the life cycle of a dog vomit slime mold. This is the very first slime mold I photographed. It was a number of years ago. It's a time in the forest when there was a cougar warning. And so the forest was really quiet, and I pretty much had it all to myself. It was great also for harvesting mushrooms, but I noticed that I thought, what is that? What is it doing? I want to know what's happening here. So over a period of several days, I took a number of photos. You can see the first one here. That's actually at the fruiting stage. And then it starts drying and separating. But you can see the slime trails here, right? These are just the coolest things. And then it just moves on. And the last slide, it's a little bit black and slimy. I'm not necessarily sure it would be that dark, but there was some rain in there. But ever since I took that series of pictures, I have been fascinated by slime molds. Okay, so what you will first notice in the forest when you start looking for slime molds is pretty much any slime mold in the fruiting stage. It's where the pathways have been reinforced to the food and is surrounded by the slime mold and eaten. You can see the slime trails or just the general lumpiness of the slime molds that wrap themselves around branches, but primarily can be found on rotten wood. It's usually deciduous, but sometimes coniferous stumps if there is enough moss cover. They'll also pop up in less hospitable environments, but they don't really thrive. But some of these, like, I mean, that's just the most awesome thing. They're just the way they look. And then, of course, that one looks like a little Pac-Man. I have made them into different things. And pogo sticks, they remind me of, they're sort of foodie, but not really foodie. And I'm going to go to the next one. Okay, so here's a couple more. Sometimes it's obvious which slime mold it is, but sometimes it's not. To 100% identify a particular slime mold, you have to wait until the spores are dispersing and do a microscopic assessment. I just eyeball them. The most common by far here in the Cumberland forest is the dog vomit slime mold, followed by tapioca slime mold, which is whiter usually, and it seems to hang out more on sticks than it does on actual stumps. But I've only ever seen them in the spring here. Slime mold season in the Cumberland forest starts anytime from early April and usually lasts into November. It mirrors the mushroom season, but conditions have to be good or you won't get good slime molds. If you see a lot of mushroom fruiting, you're generally going to see slime molds too, but not always. They seem a bit pickier when they fruit. They like moist, decomposing organic matter, but they don't want to be wet. Fortunately, the lower trails in the forest here are full of yummy good stuff for slime molds to eat. And I don't often see slime molds in the clear cuts. And the one on the far right, that looks like a bunny to me. They often have cartoonish shapes. And I just really enjoy their visual appeal. And I try not to poke, or at least until I take a photo. I just imagine that scene in Men in Black when Will Smith sticks his finger into the globe and inside is that little system and they're going, ah! So I try and be respectful of the space of the slime molds. Now, okay, next slide. Okay, so just look at this magnificent dog vomit slime mold. Look at that great slime trail, and it looks like a slime mold is perching on the top of that stump, just chowing down. And this is a big specimen. In prime slime season, walking a few trails, it's not uncommon to see 9 or 10 or even 11 slime molds just by the sides of the path. Some look like a little sticky thing thrown against a tree, and others can be as large as 1 meter across. But some are really, really tiny. you have to keep your eyes open and you'll find some amazing slime molds in the Cumberland forest. Okay, so Plasmodium can do one of two things. It can become dormant, dry and hard. It still looks like a Plasmodium, but it's no longer soft and slimy. It can reactivate into a Plasmodium when it's moist enough. And it also, of course, we now know retains memories from its previous life. This is an example of a large dormant slime mold. And there's a technical name on there I'm not going to even try and pronounce on the bottom. Okay, and here are several more examples of dormant slime molds. And of course, this one looks like a butt. So and it's all these spread out dry things. And this is a slime mold that's probably not going to be very long-lived because it's just on the gravel. It's kind of sad, actually. But they are very successful at reproducing, so I can't feel too bad for them. The other thing that Plasmodium can do is reproduce. And slime mold sex is just as weird as you would expect. If the slime mold hasn't gone dormant, the other option is to fruit and grow stalks and release spores packed with sex cells carrying 3 variable genes. Every sexually mature slime carries 2 copies of each gene, and each one of those genes can come in a variety of slightly different flavors. All the possible combinations add up to hundreds and hundreds of possible sexes. It isn't always the best way to reproduce, but it does mean there's a lot of genetic diversity in the species, making it resilient to disease or environmental changes. It can break up into small pieces and make a lot of fruiting bodies, or it can fruit as a whole that looks massive. Some maintain the shape of a Plasmodium. Fruiting bodies that are small, like a millimeter or 2, are up to a centimeter, are called sporangia. They may have their spore mass suspended at the tip of a stalk, or they may sit directly on the surface of the substrate. Sporangia that come from a single large Plasmodium can form large groups. It's only when the fruiting bodies are fully mature and the spore masses are dry and powdery that the species can be properly identified. At the slimy stage, even experts, of which I am not, they can have trouble identifying different types of slime molds. Okay, I have a series of different slime mold photos taken in the Cumberland forest at various stages of development, most at the fruiting stage because that's when they're most visible. This one was particularly interesting. It had these amazing little round balls. They looked like candy-covered cookies, but you could just see the little tubes coming up. And of course, there is a poke mark right here, but it wasn't me. And these 2 pictures, these were taken a day apart. You can see just how rapidly things change. You can see that little bit of that log there is here. And that's actually true color there too. It started off, and then I went back the day after and it had rained, so I didn't get any more, but I did see something similar on another trail a few days later, and it went kind of brown and crusty. So, but most of the slimes I seem to, they tend to go dormant rather than fruit and reproduce. Maybe it's just not a sexy woods, I don't know. Okay, so we got, this is another common one that you see. Wolf's milk slime mold. I just saw one yesterday when I was on Andy's walk, Space Nugget. It's one of the later fruiting varieties, and it has the best poke factor of many slime molds because the outside casing is a bit rubbery, and when you poke it, it oozes an opaque pink liquid, kind of like Pepto-Bismol. I've never tasted it, but it just looks like that. They're amazing. They're not very big, and they tend to keep in little separate sections. Okay, there's a couple of less common varieties in the Cumberland forest. That large black one I touched, and it felt like sandpaper, but it was big, and it was just obviously the same shape as the Plasmodium before it spored. And this one here was really, really gummy and sticky to the touch. I think it was probably insect slime mold, but I'm not 100% sure. And this is one of my favorite varieties, and I haven't seen too many of these. This is a specimen I saw on Mama Bear's Trail of Tears a couple of years ago. It's a chocolate tube slime mold in the spore setting stage when the spore mass is suspended at the end of stalks. And they look like these the cocoa covered pogo sticks, but they're tiny. They're just the smallest little things. And if you don't pay attention, you're just not going to see them. Okay, slimes play an important role in assisting in the breakdown of organic matter in the forest, which is an ongoing process. With trees, you first identify bracket fungus or conchs that signal that the tree is not healthy, followed by other fungal life like oyster mushrooms, followed by birds looking for bugs, birds making nests, bears scratching, and then you've got between these three sites, you've got slime molds. They're near the end of the cycle of returning trees to the organic matter in the forest floor. And then it's followed by my dog right here, who just digs up the forest floor after the slime molds are finished with it. So if you're lucky, you get morel mushrooms fruiting in the spring after you have landscape fabric installed. You know, that's one thing. You can find slime molds domestically within your garden, but if you're really lucky, you get a dog vomit slime too, and that's an indicator that your mulch is really nicely broken down. There are over 900 varieties of slime mold in the world, and they are everywhere except the very coldest locations. They are beautiful, they are weird, they are quirky, and they are worthy of our admiration. And that concludes my talk. Thank you very much.
Yay! Or this, whatever people want to do. Thank you so much, Dawn. That was fantastic. I can't wait because you're an observationist, so we can just pick a topic next year, and you can do like another amazing presentation. That's great. Now, does anyone have any questions? Now, recognizing Dawn, you know, expressing that she's not necessarily well, it wouldn't be a mycologist because it's not a fungus, a slimologist, but about her observations in the forest, or any feedback you want to give to her. I'm just hearing amazing presentations. So great, Dawn. Thank you. I love slime molds.
Slime molds are the
Wonderful.
best.
Yeah, they're amazing. Well, and it's been so wonderful. I really suggest you follow Dawn on Instagram, because it's just such a delight all the time to be able to see the photos that she's posting. They're just beautiful. Awesome talk. Oh, there we go. Any advice on photographing slime molds?
You know, I've heard this before, and I just use my camera phone tends to take the best photos. I have a good camera. I haven't really figured out something with a very shallow aperture. It doesn't seem to work for me in low light, but my Galaxy S8 or any good quality phone camera, you just fiddle around with it. Sometimes you have to take 4 or 5 pictures, but you're going to get something that's you're going to be happy with.
Wonderful. And Andy's there. Hey, Andy, you ask your own question.
Sure, Dawn. If you could be any slime mold, which one would you be?
Ooh, that's a good one. Well, I really like that black one. That was like, it was all crusty, and I would love to have seen what it looked like before it had spored. But probably, if I had to be honest, I'd probably say wolf's milk, just because it's just silly. It's just a ridiculous, unnatural color of pink in the forest. It's just ridiculous.
Dawn, thank you so much for everything.
You're most welcome.
That was fabulous. So yeah, that was just great. Thank you for being part of the Science Pub. I'm going to get going right away. There's a lot of yeses on the Zoom chat. To our next presenter. If some of you were lucky enough to be part of a Thursday night presentation with Jason, but if not, you're going to get a snapshot on the very important content he has. When we were first putting this inaugural festival together, some of the folks from the South Island immediately suggested Jason because of the fascinating work that he's been doing around observing death caps in the South Island and his interest in toxicity in mycology. So it's been great to get to know him, and I'm just going to immediately pass the torch over to him, and we'll get right away into our next presentation.
Awesome. Thanks. So, yeah, my presentation the other night was more focused specifically on my death cap observations in the Victoria area around the South Island. But tonight I kind of wanted to talk to you guys more generally about mycotoxins. So I'm just going to load up. So yeah, mycotoxin basics. A little bit about my interest in mycotoxins comes from my interest in which mushrooms I can eat. And I get asked all the time, can I eat this? And I thought I better get pretty good at knowing the answer to that question. So we're going to get kind of right into it here. There's various types of mushroom poisoning that are quite common and others less common, but most commonly you see gastrointestinal irritants, alcohol-related syndromes, muscarine, gyrometrin, and monomethylhydrazine, or MMH, isoxazole derivatives, mucimol, ibotenic acid, and their relatives commonly found in Amanita muscaria and Pantherina and related section Amanita species, psilocybin, psilocin, and relatives, magic mushrooms, arelanine, which is a major toxic constituent that we see in Cortinaria species, and some unknown toxins or speculative toxins that cause prompt kidney damage that we see in Amanita, in section Lepidella, and others like Amanita smithiana, as well as amatoxins that we see in the death caps and relatives like destroying angels, as well as many other unknown toxins. The most common poisoning type we see is gastrointestinal irritants. These symptoms usually onset rapidly within 30 minutes of ingestion. They're usually self-resolving with very little complications, and they may result from normally edible species. A big thing that a lot of people don't take into consideration is that everybody reacts differently to everything that they're putting into their body, and mushrooms are phenomenal chemists, and our bodies aren't always. So we're not always compatible with the same things as everybody else. So there are some practices that are good to observe to limit your risks of gastrointestinal upset, even when trying new edible mushroom species. We'll review a bit of those in a little bit here. I'm feeling a little bit underwhelmed because I don't have any really cute dogs in my presentation. I get to be a bit of the boogeyman, tell you about the scary stuff. Alcohol interactions, fitting for a pub night, I guess. Coprine is the most common, though not exclusive, alcohol interaction that we see with fungi. It causes the antabuse-like effect, and antabuse is a drug used for treating alcoholism, and there's a pretty extreme reaction within days. If you ingest coprine containing mushroom, within days, either direction of consuming alcohol, you can experience some pretty strong effects, which include accelerated heart rate, flushing and heating of the skin, and just general GI distress and upset. Other than that, we see reactions to other mushrooms, including like visual distortions and lack of motor coordination that may result from some Stropharia species and others. Even common edible mushrooms may cause gastrointestinal disturbance or distress when consumed with alcohol. I have a good friend of mine that's eaten chanterelles a lot, but we had a couple of bottles of wine, debatably too much, with a nice chanterelle dinner, and both of us did not feel well the next day. And I'm relatively positive it wasn't just the wine. Muscarine is probably the most common mycotoxin that I encounter. I deal a lot with helping people identify mushrooms that their children, their pets, or others have accidentally ingested. And Inocybe, Clatocybe species are by far the most common culprits. Muscarine causes profuse sweating in humans, severe gastrointestinal distress, tremors, excessive salivation, and neurological symptoms. In dogs and cats, you see it mostly expressed as convulsions and excessive salivation to the point of like their mouth is just running. This is a potentially serious poisoning in pets, and then even in humans in sufficient quantity. But fortunately, muscarine is one of the only mycotoxins that has a specific antidote, and that's atropine, administered by a medical professional. And there are some relatives to atropine, like scopolamine, but they have other implications for use. Gyrometrin and MMH, or monomethylhydrazine. It's a volatile compound, and what I mean by volatile, or as an example of what I mean by volatile, is they use it in satellite thruster fuel, and there's a component in rocket fuel. So it is thermally sensitive. It can be destroyed by heat, and because of that, it has been used culinarily through a process of parboiling to remove the toxins. However, good ventilation is required because gyrometrin and MMH can become a harmful vapor, and in some situations in Europe, this has resulted in the chefs preparing it losing their lives. In severe cases of poisoning from gyrometrin and monomethylhydrazine, liver, kidney, and red blood cell damage may occur. And in trace quantities, gyrometrin and MMH has been shown to be carcinogenic. Isoxazole derivatives, mucimol, ibotenic acid, and relatives. We all recognize this mushroom. Amanita muscaria is probably the most photogenic and well-known mushroom. I actually think Shannon used the same photo in her presentation. I took it from the STIMS website. Don't tell anybody. This mushroom has an amazing history in cultural use in parts of Europe and Asia, and some experimentation and recreational use in North America. And while deaths are rare in humans, they're not entirely unheard of. And we do see more severe reactions in pets like dogs and cats. While a lot of people associate this mushroom with hallucinations and the magic mushroom, the experience is generally not that pleasant. It's pretty disassociative. It can result in gastrointestinal upset, profuse sweating, convulsions, and general illness. And it has resulted in limited fatalities, though usually in combination with other compounds ingested. Psilocybin, psilocin, and relatives, your classic magic mushrooms. These are relatively safe physiologically speaking. The LD50, or like lethal dose per 50 kilograms, is like astronomical. You would have to eat essentially your body weight in psilocybin mushrooms to be in any physical danger. However, you're going to be experiencing some pretty profound effects in the meantime. Greatest danger lies in misidentification, and that's why I've included these two photographs in this slide. I hope you guys can see my cursor, but on this side on the left here, this is Psilocybe cyanescens, and they're a local native Psilocybe species. And on the right here, we have Galerina marginata. And these are amatoxic mushrooms. They occur in very similar habitats. As you can see, they're both growing on woody debris here. They have very similar cap coloration depending on their environmental conditions. And it would be a relatively simple mistake for a novice to make. So this is one of the last mistakes that you want to make, or potentially would make when it came to mushroom identification. So the point of this presentation, this kind of really hits it home, is not to scare you away from mushrooms or make you afraid of what mushrooms can do. Psilocybin's a great example of what mushrooms can do for us. But this presentation is meant to really hammer home the importance of having well-rounded ID skills or the ability to consult before you ingest any mushrooms. An ounce of prevention is a pound of cure. There's some really fascinating and ongoing research into psychotherapy and addictions treatments applications with psilocybin, psilocin, and related compounds, including some very well-connected companies here on Vancouver Island. Next, we'll talk about aurelinine. Found in certain Cortinarius species. This is a really dangerous toxin for kind of the same reasons as we saw and we discussed in my death cap presentation the other night. The symptom onset of this toxin is very delayed. It can be deadly, and it directly affects the kidneys. Symptoms may not manifest for up to 3 weeks. And the big danger there is that that's a significant portion of time, to the point where you wouldn't really associate, oh, I'm sick from those mushrooms I ate 3 weeks ago, with your illness, which makes it very difficult to diagnose and very difficult to treat medically. Around 8 days is the average onset from ingestion to symptoms with our aurelinine ingestion. We don't see a lot of aurelinine containing quartz, Cortinarius, in our region, but there are some out there, and the whole large and very diverse genus kind of gets overlooked a lot just because of the few species that are known to be aurelinine toxic. Next, we're going to move into amatoxins. I covered this pretty thoroughly the other night with my death cap presentation, which I hope some of you were able to attend. The delayed onset, again, is a major factor in the risk and treatment of amatoxin ingestions. The severe effects can take many days to progress through the symptom course and ultimately result in a fatal outcome. Amatoxins affect the liver, the kidneys, and clotting factors in the blood, and can essentially make you bleed out through your internal organs. There's been some trials in the United States with a compound called psilocybin, which is an extract from milk thistle and used in early stages of Amanita or amatoxin ingestion for protection of the liver. My understanding is that those trials are currently over and that it's not readily available. So these are definitely species you want to be familiar with. They're found in Amanita section Phalloideae, which is the death caps, the destroying angels. And I'll talk a little bit about destroying angels here. So destroying angels is the common name for the all-white Amanita related to the death caps. And with all the media attention that death caps have been getting on Vancouver Island, we see a lot of people very concerned about it. And it's a big part of why I built the other presentation just about death caps, is to try and raise awareness and reduce anxiety. I don't know about the others on the panel, but I get a ton of people messaging me saying, "Hey, I think I found death caps in my lawn," and they're very concerned. And I think I found a destroying angel in my lawn. I've not seen any reliable documentation of any of the destroying angels on Vancouver Island. And 99% of the time, somebody sends me a photo and says, "I think I have a death cap." They have Leukocaricus Leukocytes, which is an all-white mushroom with white gills and white spores. It has a ring on the stipe, and it has a slightly bulbous base. But it is not toxic. It's quite common. And it's probably responsible for, like I said, 90+% of the reports of death caps that I see on Vancouver Island. We also, I don't know how to go back. We also see it in Galerina species, Lepiota species, and Foliatina species, which are all relatively common occurrences in gardens, in lawns, and just in general in the forests around here. So amatoxins aren't anything new to our local area, but death caps are. We are on the lookout for them, and all the time new information's coming in that's helping people like me find them. Like Shannon and Paul Kroger from UBC had a great paper that documented the mycorrhizae with Gary Oakes, and that's kind of expanded the areas in which I'm searching for them as well. And yeah, so what else is toxic about mushrooms? There's a lot of unknowns when it comes not only to toxins in mushrooms, but to which mushrooms are toxic. So many mushrooms have unknown edibility or toxicity information. There's just there hasn't been a lot of research into them, or they don't have a long established record of being safely edible. Others are known to have toxic effects, but we haven't established what toxin causes those effects. Sometimes we're just not sure why some mushrooms are toxic, or why some people will react a certain way to mushrooms when most people don't. A great example of that is the chicken of the woods is a very commonly eaten mushroom, and it's seen in field guides a lot as a choice edible species. Lidoporus, they are known to cause a disproportionate amount of gastrointestinal incidents, and in some cases quite severe incidents. The North American Mycological Association even records a fatality, which is not something you would generally expect to see with a mushroom that's considered a choice edible in virtually every field guide. There's been a lot of speculation about why that might be, if it's related to the host tree species or not, but we don't really know what causes that. But approximately 10% of people experience a strong GI type reaction to Lidoporus, regardless of what it's growing on. So some good pointers to avoid an unfortunate experimentation with mushrooms is avoid experimenting with mushrooms of unknown edibility. If it's not known to be an edible species, you probably don't want to be the guinea pig. Practice caution when you're trying new mushrooms for the first time. Even with common edible species of wild mushrooms, caution is always good. So a good practice is to maintain a portion of your collection for identification should it be necessary. Start with a very small amount and wait 24 hours before you ingest any more of it, just to see how you personally react. I hear a lot about people that have played around with experimenting with edible mushrooms and had some unfortunate incidents. So that's kind of led me into part of what I do now. I have a big focus on mushroom identification, and that's kind of led me into identifying mushrooms on a volunteer basis for organizations like the ASPCA, the Pet Poison Line, and Poison Control, who don't always have identifiers readily available. Good pointers are definitely to take some identification courses, attend workshops with like some of the people presenting tonight, join your local mycological society and listen to the lectures, and attend events like this every opportunity. You'll learn a ton. I wanted to go back to one of my slides here because I kind of accidentally skipped over this one. So prompt kidney damage. I don't know why this one got skipped, but it was one that I really wanted to cover. Amanita smithiana is our local member, our most prominent local member of section Lepidella and Amanita, and it can cause some quite serious kidney damage. It's commonly misidentified as a matsutake or pine mushroom. And as little as 6 hours after ingestion, it can induce some pretty drastic kidney damage or kidney failure. Treatment can require long-term dialysis and extensive courses of medication. So this picture here shows a pine mushroom and a Smith's Amanita. I have been at festivals and held one of each up in hand, or even held them out to people to examine and ask them to tell me the difference. And the number of people that can't tell the difference between Smith's Amanita and a pine mushroom is concerning. You definitely want to be aware of this mushroom and its relatives if you're going to pick pine mushrooms, especially if you're going to eat pine mushrooms. Last year, there was a gentleman in Whistler who was given some pine mushrooms that didn't turn out to be pine mushrooms and caught a flight to Toronto and was promptly moved into ICU upon landing. That's kind of an example of how fast that toxin can act and the severity of that ingestion. So yeah, the long and short of this is that mushrooms are phenomenal chemists. They've been at the game of producing organic compounds for a very long time, and they're really, really good at it. There's a huge amount of takeaways that we can take from toxic mushrooms. Like, what a mushroom produces as a toxin can have applications to us in the context of magic mushrooms as one example for psychotherapy, for addictions treatment. What a mushroom produces as a toxin in minute doses can be used to treat other diseases. For an example, even in some of the amatoxins in Amanita phalloides have been examined in cancer research. We're also seeing like drugs like sorry, what am I drawing a blank here? Antibiotics. Sorry, it's like how can I forget antibiotics? Coming from fungi. So the value in understanding toxic mushrooms, not only their risks, but really taking a hard look at their chemistry is I think we're kind of one of the frontiers of mycological research is going, and I'm really excited to participate in it. So yeah, that's my toxins presentation in a nutshell. I'm happy to take any questions.
We have one. Thank you so much. Let's do the clap thing so that we stay interactive. Woohoo! We got a question from Jason. Curious if cumulative toxicity over the years or decades is a thing. He seems to have become allergic to all mushrooms after years of enjoying them.
You can build up sensitivities to anything, really. Consistent exposure will do that. I'm not a toxicologist by training, but like we have seen examples of that, and there are toxins that do have cumulative effects. Like I didn't mention in this presentation, but a common mushroom that we see around here, Paxillus, known as the poison pax, for a long time was classified as an edible mushroom. But regular ingestion of that will result in a autoimmune effect where basically your immune system recognizes your red blood cells as a pathogen, and it's an ultimately fatal condition. So we don't understand a lot of this chemistry, honestly. So yeah, like there's entirely an avenue for continued exposure to result in a sensitivity for sure. Cumulative toxins, I would kind of associate generally with like bioaccumulation, which is another kind of aspect that you can see with fungi, though it's not as common because you're relying on like pollutants in the environment to bioaccumulate in the fungus itself. Heavy metals being one example. Leukocaricus species are really effective bioaccumulators of heavy metals, and so like ingestion of that's kind of sorry to go off on a tangent there, but that's kind of like what I would generally associate with cumulative toxicity.
In the actual mushroom instead. I have a quick question I want to throw in, just because I spend a lot of time with kids in the forest and have had this as a general practice of asking of especially if I'm with like a classroom, like I'm helping them go for a walk, around not touching the mushrooms.
Right.
And so they were curious even to use a stick, but that's not based on knowing if there are any mushrooms around here that would be dangerous to touch. Like, I'm not worried about them eating it, but I'm trying to be careful.
I hear this a lot, and at the end of the day, when we look at some of these compounds, you can absorb some of these compounds through your skin. In the levels you're likely to find them in any mushroom, there's no danger in handling any mushroom under any normal circumstances. You know, if you have a large open wound on your hand, basically don't crush mushrooms up into it. But there's little to no evidence suggesting that there's any real danger in handling fungi. And as an example of that, the taste and spit test is often used in mushroom identification. So even with toxic species, nibbling a small amount of it, chewing it up, and then spitting it all out will give you some interesting characteristics, but you're not reaching a threshold dose. Like with any toxin, you need to have sufficient dosage, and with mushrooms, you need to ingest a significant quantity before you're in danger. Like, what constitutes a significant quantity might vary amongst these many different mycotoxins, but your threshold dosage is an important factor. And that's part of why we see more serious poisonings from some of these toxins in pets than we do in people, just because, like, not only does their physiology work a little bit differently, but they have less mass, which plays a big role in how you're capable of addressing those compounds.
Awesome. I'm going to do grab Kay's closing question and kind of lump it together with the takeaway from you. In classic Cumberland Forest programming tradition, we're of course going over time. So I just want to. I'm not even
notorious for that. Sorry.
No, it's not. It was cumulative. It was a cumulative process that we're all part of. So it's okay. But Kay had asked resources that you would recommend for foragers that want to be more confident around identifying toxic lookalikes. I'd like to ask you that, and also just any takeaways that you want to leave us with for folks that are interested in this topic and being engaged.
You know, like I said, you cannot underestimate the value in joining your local mycological society. SVIMS and Van Myco are both great examples where you have brains like Andy's and Shannon's and the Jessicas and others to draw upon. There's a huge value in attending those talks and participating in those club meetings, as well as we have a great amount of local social media groups and less organized enthusiast groups that have a lot of knowledgeable and experienced people. Getting some good field guides and familiarizing yourself with them is probably your best practice. But you know, there's no shortage of experienced people and no shortage of availability to attend events like this and actually talk to them and learn the ropes. I know a number of our locals host and offer workshops or classes on individual or group basis. So yeah, I mean, it's, if you want to spend a little bit of money, then definitely take some courses. It's well worth it. Ultimately, you want to be 100% sure with whatever it is that you intend to eat, because it's going to be you that has to face the consequences of that decision, and not the person that told you that that was a pine mushroom.
So Kara has posted in the chat the link to Jason's group as well, which helps with mushroom identification and toxins identification. There's Dawn's clarified a few others. Jason's been really great on the chat too, so if you had any other questions for him, you could fire them over on the chat if you like. But I would just like to thank you so much. I know that it was a very busy week for you, and I'm very, very grateful that you were willing to share time with us. And we look forward to having you in Cumberland in the flesh in the future, because it would be really great to have you up here in that future time when we get to do stuff together.
Well, I very much look forward to it. I am actually looking forward to the rest of these awesome presentations.
Yeah, no kidding.
Now I'm regretting I didn't put any dog pictures in mine now, though.
Well, next time. You're learning, right?
Yeah.
So I'm totally canceling the break. So if you need a break, you can just like run to the bathroom or get your drink. We're going to keep going. We've got 2 more phenomenal presentations, and we want to, we want to give them our all. So we're just going to keep going. Pretty excited to welcome our next presenter for a bunch of reasons. Yarrow Willard is a clinical and master herbalist, 2nd generation, co-founder and formulator of Harmonic Arts. And if you don't know Harmonic Arts, they're a business that's based out of Cumberland, an incredible employer and business leader in our community as well. And Yarrow and his partner Angela have been really amazing supporters of the Cumberland Forest Project. And Yarrow is also an amazing innovator and international speaker. He's a director on the Wild Rose College of Natural Healing, an educator and a health ambassador who's in demand for speaking events across the country and around the world. He also does things like comes out with me with a group of kids into the forest and teaches them about the mycelium network. So he's a real gift to the Cumberland Forest community as well, and to our community at large. Always deepening our connection with the natural world through the wisdom that he brings and the teachings that he's able to share. Feel pretty privileged to call him a friend, and really happy to welcome him as our next presenter.
Thank you. Thank you. Wow, Jason, that was awesome presentation, just by the way. I really appreciated it. But it was so down, so I have to tell some mushroom jokes. I just feel like we need to just lighten up for a second. So I'm gonna ask you guys this. What did the young puffball say to the old puffball? Quit being such a spore loser. Just kidding. What did the old turkey tail, though, say to the young turkey tail that was growing above him? Quit making so much bracket up there. And if you have a reishi or a turkey tail, you know what they say to each other? I think it's time for us to take a selfie. All right, there's a couple of bad jokes because I'm a fun guy and I know some of you are fun gals. And you know, if there's one room in the house that's really small, you know what it is? It's the mushroom. Anyway, okay, so I could talk to you about a whole bunch of different things. I kind of decided to pare down my presentation a lot and was just sort of playing with some slides as we went. And I think I'm going to talk to you more about medicinals. Right now, what I have behind me is a giant reishi. This is the most famous medicinal mushroom of all time. This mushroom has been enshrined in the Forbidden City in China as the emperor's mushroom. And so I'm going to talk a bit about that. I'm going to talk about polypores, and I want to talk about conchs and this whole family of medicinals. I think this is a really valuable piece in what we're learning about mushrooms right now, and it's really growing the awareness of mushrooms as medicine, especially in the modern age when we have a lot of issues around immune system regulation. And so, you know, one of the things that mushrooms do that is amazing is many of these polypores work as immune modulators, which means if you're lowered immune, they bring it up. If you're high immune, it brings it down. So autoimmune conditions or colds and flus. And it's that time of year. And we have some of the best polypores here in BC. So we're going to talk about tree-loving mushrooms. That's what we're going to talk about today. And I just want to make sure that you guys are ready to go down the rabbit hole with me. What I mean is bending reality. We're going to take thousands of years of understanding from Chinese medicine perspective, from human evolution perspective, and bend it into this next 20 minutes minus the 5 minutes I've already rambled in. So let's get going. Most people, when I talk about mushrooms, they think of this one, right? And we've already seen pictures of this. This is the exact same slide you guys were showing earlier, this mushroom, except for I'm high while I'm watching it. No, but really, this mushroom is classic. It's the emoji mushroom, right? It's the Santa Claus mushroom. It's the famous psychedelic gate sensory perception shifting mushroom. And I know that Andy's going to talk a bit more about these sensory perception changing mushrooms, and that Jason already talked a little bit about that. So I'm not going to take much time other than this has really iconified the kind of folklore. And when I talk about mushrooms, people think I'm talking about this one or this one. This is what people always think. Oh, you're into mushrooms. Yeah, I'm into mushrooms too. I'm a fun guy, right? Wink, wink. No, we're not going to talk about those, although I want to say that some of the new research coming out on these around microdosing and around the concept of bending our sensory gates to perceive a different reality has some merit, especially around end of life, around depression, around PTSD, around these kinds of things. There's some really cool science coming out on this. And you can learn more on your favorite Reddit channel if you want to go down that rabbit hole. But we're not going to go down that one either. Tonight, okay, we're talking just brief overview. Mushrooms are everywhere. They are all around you all the time, from the stratosphere to a mile below the ground. So they're everywhere. They're up to a quarter of the biomass of our planet. Between mushrooms and bacteria, or not mushrooms, sorry, fungi and bacteria. And we want to get this straight. Fungi are not mushrooms. Mushrooms are part of the fungi family. They are the fruiting bodies. The ones that produce fruiting bodies are the mushrooms. But in the fungi family in general, they make up a huge piece of the biomass of this planet. Between them and bacteria, they actually rule planet Earth. Not humans. It's the mushrooms, it's the fungi, and it's the bacteria. And they are some of the most intelligent beings on this planet. They have a lot of really interesting, cool things that I'm not even going to go into, but I'm doing a talk tomorrow night, and you can join me there where we'll go deeper into mushroom mysteries and a lot more of this stuff. Anyway, they've been around here since the beginning of time, pretty much. There's some thoughts and beliefs that potentially our planet was seeded by interstellar space mushrooms that came in and crunched up the lava rock and turned it into soil. They've been on the Earth's surface for at least 1.3 million years, although there's some new petrified mushrooms that are saying up to 2.4 billion years, we might have seen some kind of fungi on this planet. They broke down the lava rock to create soil, to create the Cumberland forest, and 700 million years ago, that's when plants started to slowly get onto the Earth. So long time, plus the biggest species on the planet, the honey mushroom. We're talking well over, well, we don't even really know how old it is, but over 2,000 years, definitely older than Christianity. Okay, I'm gonna, it doesn't want me to do that. I have to click my button. So I'm gonna, I put this slide in because something I wanted to just mention is that we really don't know. We don't know very much at all about mushrooms yet. We know about 10,000 species, and yet this could only be maybe only like 10% of them that produce fruiting bodies. And if 5% of the unknown mushrooms produced some kind of medicine, then we're talking about thousands of species of unidentified mushrooms that could be producing medicinal benefits for the body. We know about 270 that produce medicinal uses. So this is, to me, the biggest reason not to cut down forests is really that we just don't even know yet. I was in the forest last year and I found this mushroom and on a trail I had been on for years walking down, and I had never seen it before. And I was like, I walked this trail for like I lived in Cumberland for 10 years and I've walked this same trail, never seen this. Just the right type of environment, the right type of moisture, and boom, it produces fruit. Some of these will go without fruit for a long time. Anyway, we don't know. That's what I'm really trying to say, is we really don't know. But we do know that they hold this, as Andy said in his talk today, the wood wide web, this nature's internet, this kind of connection of the mycorrhizal communication between the plants, between all the different species. We know that no plant can really survive very well without this fungal relationship. And we know that they aren't just, like, they're smart. The thing that's so crazy is that we look at the carbon sequestering in the forest, and we see that more carbon is sequestered underground than above ground in the trees. And it's all like, I think of the mushrooms as like the Swiss bank account of the forest. They're holding all this cash, and they've got all that energy. So beyond that, I don't have time to totally talk a lot about nature's internet, but the intelligence of the wood wide web is astounding. The more we learn about it, the more we see how mother trees feed their young via the mycelial nets, the more we see how the communication goes around the forest between what predators are there. Some of the cash that they share in the ground is then given to various trees that are having a hard time that year to create more biodiversity, ecosystem stability. The concept here is that mycelial nets essentially grow forests in order to create a stable environment where they can be the Swiss bank account to hold the cash and to create the right environment. All right, what I am going to talk about tonight is tree-loving polypores. And I call these, I like to make up my own science here. I call them the ennobled species and the levity eaters. We were joking before about this mushroom that's behind me and how like this anti-gravity effect, or like how the gravitational pull shifts the way in which the mushroom shapes. Well, I like to think of all like walking humans and all plants and all fungi, well, really the fungi are the eaters of them, as levity producers. We defy gravity. We walk upright. Yes, gravity affects us, but if we know anything about the yin and the yang and the night and the day and the male and the female and the black and the white, we know that there's this polarization. Even if you look at Facebook, is there polarization? Yeah. There's always going to be polarization. So what's the opposite of gravity? I consider it levity. And I think that the trees produce the most levity of any species. They grow hundreds of feet in the air, pumping blood, defying gravity to the best of any species on this planet. So the smart mushrooms, the evolved, ennobled species, are the ones that eat the trees. They eat the trees because the trees contain the most of that kind of upward moving vitality, that vital energy. And you are a levity eater too. You eat the levity, the fifth element, the vitality in the food you eat that gives you your vitality, that gives you your levity. So bear with me with this term levity, but this is what I like to think of them as. And all of these polypores produce the most profound chemistry. They produce a whole group of, and there's a couple more, we'll talk about some of the individuals, but they produce, wait, let's go back. I'm gonna go do this slide, and I'll come back a couple. They produce polysaccharides and triterpenes. These are two really cool groups of chemistry. Now, they are trapped in these fibrous cells. So in order to extract them, we have to do different methods, and I'll talk about that too in a minute. But essentially, there's chitin and lignins. Lignins are like plant fibers that are in the trees, and chitin is like the exoskeletons of insects or your nail beds or your hair follicles. These are essentially these different compounds that are held within that structure. There's also some polyphenol groups. There's also some sesquiterpenes in some of the more fleshy mushrooms like the Hericium family and some of the oyster mushrooms. They've got some of those sesquiterpenes, but the triterpenes are a lot of the really potent chemistry that we see in these fruiting bodies, these mushrooms. So I'm going to go back a couple of slides, but really what I'm trying to say is that this chemistry is unlocking some of the most unique characteristics in us, and they use these compounds, these beta-glucans, these branched polysaccharides, and these triterpenes for their own defense system. Think of these medicinal mushrooms as hanging out in some of the harshest environments on the planet, right? There's all kinds of other predators, bugs, viruses, bacteria, pathogenic organisms trying to get in to feed on the tree that they're feeding on. And you will see that many of these species will share a tree. I've seen turkey tails on the same tree as I've seen artist conchs, or I've seen like Ganoderma Organensis or Suja on the same tree as the artist conch. So we will see a lot of them grow together, but they're using these compounds as their immune system. And that's where the key is to how they affect our immune system. Okay, so I'm going to go back to this one. There are a lot of polypores. There are a lot of tree-loving mushrooms that grow here in BC. Again, my slide is small, but you're getting a recording, so if you want to pause it, you can pause it. These are some of the ones I've just mentioned that I've seen in our forests and around BC. We don't really have birch, so we don't see a lot of birch polypore, but every single one of these mushrooms shares something in common, and it's branched polysaccharides. They all contain some degree of these polysaccharide compounds, and then some kind of steroidal compound like the terpene groups, like the phenolic compounds that we might see in some of them, the polyphenols, the triterpenes and the sesquiterpenes, and even the diterpenes we see in some of these. There's something like that's in the fruiting body. These are like hormonal structures, whereas the polysaccharides are more like these Aztec glyph kind of huge groupings. Just as a sort of thought for you to kind of think about with polysaccharides, imagine you were at a concert and you were sitting in one chair. Well, one chair is similar to one carbon in a polysaccharide. We're talking about thousands and thousands of chairs in one chemical makeup. And what we learned about this kind of chemistry is that it's actually more like the shape than the actual ingredients in it. And I'll go back to a couple of slides. Oh, I want to talk about OTC. I'm sorry, I'm skipping all over the place, but we'll talk about polysaccharides for a second. So what's so cool about this shape is that our body reads chemistry. And this is a big hit I've got from studying the forest, from hanging out in nature. It's that when I put something in my mouth, the organoleptics of my sensory organs tell me all about the chemistry. My body reads chemistry, just like you're watching this, and maybe, hopefully, I hope you're learning something. Maybe you're not. At least you're being entertained while you drink your Cumberland growler. But what the polysaccharides do is the shape is so unique, and there's so many different structures that fold around these carbon chains that they create these really big, cool shapes that our immune system and our digestive system use to kind of calm it down and educate it. This is a college education in chemistry, and these are some of the oldest species on our planet, some of the most ennobled species that have learned how to develop this type of defensive mechanism to protect them from pathogens, and they impart that wisdom onto us. This is why we see such an upturn in people using reishi and lion's mane and chaga mushroom and all of these kind of medicinal mushrooms, is because the effect they have on the body is like educating the immune system, giving it a higher level of function, but they also work really uniquely in the digestive system, calming down the nervous aspect of the digestive system and being very anti-inflammatory. This is where our microbiome is, and this is where a lot of the health originates in our body. So we see this, as well as what we see is that almost every one of these medicinal mushrooms that have these branched polysaccharides are entirely anti-pathogenic, which means most of them are antibacterial, antifungal, which is really interesting. They'll kill candida and other fungus in the digestive system. They're also antiviral, many of them, many of the fruiting bodies, especially the terpene contents in the fruiting bodies, which is mostly found in the spore pads of these conchs, is very antiviral, which may be useful for some of you if you've ever watched the news lately. But anyway, there's also a lot of antioxidant compounds in them. What I'm really trying to say is they are a really unique building block for us to work with. I want to go back to Otzi. I'm going to just talk about him for a second. 5,300 years ago, Otzi lived in, we don't even know what kind of society, but what we do know is he had some really interesting different types of crafted tools, and he was carrying a couple of polypores on his belt. We found out that the polypores he had were really good for antibacterial property, plus they could tell from his digestive system that he had Lyme disease and he had parasites. And both of these polypores, the tinder conch and the birch polypore, I think it was, were useful for helping support him in this. So we see that medicinal mushrooms from the conch level have been used since well before our current history. We also see that many of these, like in China, for example, this reishi mushroom, which is the one that's right behind me here, has over 4,000 years of history, clinical history. They've been using it as part of their medicine path for a long time. It's been written about for well over 2,000 years. In that part of the world, they consider them, some of these medicinal mushrooms to be their most potent medicines. They contain the shen of the forest, the spirit of the forest. One more thing about Otzi that was really cool is he had some tattoos, and they actually found that he had tattoos on acupuncture points that were linked to helping you with arthritis. This was really cool because there's a concept around energetic awareness that we believe when we're a little more connected to the natural world, we can all read these patterns. And so yes, acupuncture and meridian channels and all this kind of stuff comes out of Chinese medicine, but we see in an ancient culture where Otzi was part of, that somehow they understood these various patterns in the energetic flow. And just for one more quick hit, all disease comes from stagnation. If you have stagnation, you create inflammation, friction, and disease on every level. And so what we're seeing is Otzi knew this at some level. He had stagnation, and he had literally tattooed parts of his body to help him work with that. Okay, back to our main story. When it comes to working with these polypores, they are hard. And actually, I'm going to jump back one more slide. Remember what I talked about was the varnish conch, the artist conch, the turkey tail, the oyster mushroom, which is not really a polypore, the agaricon, which is our very rare mushroom that we actually have here. Do not pick it if you see it. I know who you are. I watch that mushroom. I hang out with it all the time. I go and see it every year. Anyway, there's a few of them, but just don't pick them. The red-belted polypore, the tinder conch, the split gill, and then the bear's head is another one that has polysaccharides, but really the conchs, like the willow bracket, that's another one that hasn't been looked at much here, but we do have some willow, and I've seen it a few times, and it's a Physarum species that has really potent antiviral and anti-mutagenic properties, but most of this comes from their terpenes and comes from their polysaccharides. Okay, how do we get these out of them? They have hard shells. These are wood-like mushrooms, so you can't just eat them. You know, I've got a mushroom here. If I try to eat this thing, there's no way. There's no way I'm going to eat these mushrooms. So I got to break out the chitin and the lignans, which means I got to decoct them. I've got to simmer them down. So the first method you want to make as an extraction is you're going to make a decoction. You're going to lightly simmer it like a big pot of tea, right? But you want to kind of simmer it. You're not going to just brew it up like a tea because you're really got to break down that layer. In the water, you're going to get your branched polysaccharides and some of your phenolic compounds. Some of them will evaporate, but you're not going to get your terpenes. So you need to make an alcohol extraction as well. So really, if you're going to make mushroom medicine, what I have here on the bottom of this picture is me with a few of the mushrooms I've harvested. I'll slice them up and I'll make a decoction to begin with. And actually, I just happen to be drinking some chaga tea, a mushroom decoction tea. Right now, I drink it all year or all fall. I'll add into this a little bit of our local turkey tail, a little bit of our artist conch, maybe some red belts. I don't like the red belts as much because they're a little strong in their ergosterols and their phenolic compounds that can be really strong. So I don't use too much of them. But what you want to do is you want to simmer it down for at least 20 minutes to an hour to 5 hours, brew it down to a nice thick liquid, and that'll last for a long time. Many of these mushrooms are also antiseptic, so you're going to get some great benefits out of the antiseptic qualities, and they stay in the fridge or on the stove for a long time. You can also make dual extracts into a tincture. One of my favorite ways to do this is to take your decoction, freeze it, and then take all the leftover marc and put it in a bottle of vodka. Then you put the two together, voila, you've got yourself a mushroom tincture. Or you can do it the opposite way where you make an alcohol extract and then you put it into a decoction, put the two together, you got all the best chemistry. Another way to work with this is a decoction syrup. What this is to make a hot decoction like your tea and then add some honey. This is a great way to work with kids. So I'll do honey and water essentially just to make a nice syrup. If you do 50% tea and 50% honey, you're going to get something that can stay in your fridge for a number of weeks. Easy to work with your polypores. Most of the polypores you see in the forest are on that list of that I put earlier in the slides, and almost every one of them is easy to recognize and is a beneficial immunomodulating medicine to make into a decoction. But don't be going out and picking all the forest mushrooms. Okay, so here's another kind of picture of making it like that, slicing them up, decocting them, making the alcohol extraction, putting the two together. What I do want to talk about is, here's one of my favorite mushrooms that's in our part of the world. This is the Ganoderma Organensis. I love this mushroom. It's an annual mushroom. It fruits every August and September. Just a really potent mushroom. This is 1 year's growth. And to me, that denotes a lot of vitality. It's our most closely related mushroom to the reishi of the Orient. It's our spirit forest mushroom. This mushroom in China, this type of mushroom that's an annual that grows like this, is considered meditation in a bottle. And for me, I don't even pick this mushroom much anymore. I just like getting out, and I like to lick the spores off the top. This produces a ton of spores. You can get out and just lick the spores. You can see in the far corner picture, I have a hand with spores on top of it, and I'm licking them. One of my favorite things to do. Powerful spirit mushroom, grounding the body, great for ADD, great for just bring us back into our body. We use this for things like calming down the immune system, but also calming down heart palpitations, calming down the breathing, all these kinds of things. Anyway, great mushroom, love it. It's out there. It only fruits once a year. If you see it, give it some love. Next one I want to talk about is artist conch. This is the mushroom I have right here. This is a really big artist conch, but this is a very common mushroom in our forest because we have these beautiful trees and they grow some artist conchs. This one releases, get this, 5 trillion spores a year. You think there's a lot of people on Earth? Well, one mushroom has way more spores, 50 or 30 billion spores a day. And at the right time, when the sun is coming down at the right angle, 21 million spores a minute. This thing just, and I've tried to take some photos of that. It's pretty cool. I definitely think that Shannon or Dawn is going to enjoy trying to get some pictures on her phone with that, of the polypores as they spore. But what's cool about this one is it's also something that's been studied to be used in animal feed. And you can actually take this, inoculate poplar wood shavings, and produce a digestible animal feed. I think this is far better than GMO corn, and I wish we would do more of this for feeding our livestock in the world. It's a great food source. In fact, a lot of these medicinal mushrooms, I went to a medicinal mushroom conference last year in China, and there's a number of studies around inoculating wood with mushrooms and producing edible food stock that was more immune enhancing for livestock. So I think this is a great future topic to have somebody kind of who's looking at this and who's studying this. I think there's a lot to the benefit of working with medicinal mushrooms in our animal husbandry, and a little more of that. Okay, another thing that's neat about this one, the reason it's called artist conch, as you can see by the bottom picture I have, I took a feather from my hat, I think I got a feather here, and I scribed it in and wrote our Harmonic Arts botanical dispensary. You can make art with this one. It's beautiful. This was traditionally used by many First Nations cultures to produce signage that said, hey, camp's over there, or we left 2 weeks ago, or whatever that might be. The other thing was, it was used in this part of the world by many of the First Nation cultures as a smudge for mosquitoes and black fly. It was also used for mouth infections and excessive saliva. This is something that was one of the traditional mushrooms. I know there was a question earlier about, hey, have the truffles been used by First Nations cultures? Unfortunately, we have lost so much of the native use of mushrooms. And from what I've understood from some that I've heard, there was a bit of a fear of some of the mushrooms, but the ones that were used were polypores. They were often, both this one and the red-belted polypore, were put into soups as a preservative, as a refrigerant, to keep your soup from going bad for a long time. So especially with meats, they would be used to cook up in the pot to keep it from going bad. Anyway, also in Africa, Jane Goodall was looking at this mushroom, because this mushroom grows all over the place, and the gorillas loved this mushroom. These big, robust gorillas would nibble on it, and obviously the alpha would take the mushroom from the omega or the beta and say, no, my mushroom, hang out with it. They would covet it and eat all the edge, all the spore pad of these mushrooms. So if the gorillas are eating it, it creates better digestibility for animal feed. Hey, there's something to this. This is an anti-tumoral, antiviral, antibacterial, anti-diabetic, anti-inflammatory mushroom that is also a very profound and profuse grower in a good, intact ecosystem. So it's been studied for all kinds of things around that immune modulating, immune regulating, working with Hashimoto's, working with a lot of these different things around digestibility, inflammation, pain. And that's all the time I have because I want to talk about a couple more. So here's one that you're going to see everywhere in your forest. This is the most hard-working polypore on the planet. I got to give big love to the red belt because it's usually the underprivileged, underappreciated. You will see lots of these that don't have a red belt. And this is probably the most common question I get around polypore. It's like, oh, what's this one? And like, a red belt. Oh, what's this one? Oh, a red belt. Oh, what's this one? Oh, it's also a red belt. It just doesn't have that bright red belt. You see this everywhere. So this mushroom was used as a tinder mushroom to carry fire. This is was a powerful fire-carrying mushroom. It was also used as a smudge, similar to the artist conch, but it's got potent antibacterial properties. One of the ways it was used by First Nation cultures was powdered and put into dress wounds. So this was a wound care mushroom because it's got antiseptic, antibacterial properties to this one, and antifungal and parasitic properties. It's a great one for that. It was drank as a tonic for inflammation of the digestive tract. This was a common way it was used. It was also smoked by First Nation cultures for headaches and headache remedy. This one's easily added to soups, and it's not my favorite, again, because it has so many steroidal compounds that have really actually a nice smell. If you smell the underside of this, it smells like watermelon rind. Really cool mushroom. Like I said, it's the hardest working mushroom in our forest. So when you see it in the forest, my suggestion is you give it a little pet and you say, hi, red-belted polypore, I appreciate you. And also something you'll see, there's something called mycopee. I know a lot of you have seen it. It's the sweat on the mushrooms. When it's a hot day, the mushrooms will be sweating as they're holding that. Drink the mycopee off of the red-belted polypore as a way to pick up its essence. There's something that's really potent about tuning into a very small amount of the energetic, and your energetic body becomes much more hypersensitive. So drink the mycopee when you go by the red-belted polypore. It's a wonderful way to connect with this mushroom. Okay, this one also contains a lot of, like I said, essential oils. It's actually had antihistamine effects, so calming down allergy response, not just in seasonal allergy, but allergies in the digestive tract. So again, another one you might want to add to your teas if you're brewing something up. Okay, next one, and I'm only going to take a few minutes. I'm going to probably pause it here quick because I could talk for a couple hours, but you can catch me tomorrow night talking for a couple hours, or at least an hour, and as long as they let me talk for. But this is turkey tail.
You can go as long as you want tomorrow night, Yarrow. Totally.
This one's everywhere in our forest too. Another hardworking mushroom. What's so cool about turkey tail is that it has the most research of any of these mushrooms. The only one that's been researched a little more is reishi, but this mushroom, like we're talking hundreds of millions of dollars have been spent looking at turkey tail, and it grows all over our forest. It grows everywhere. It's another hardworking mushroom. There are some foolers though. There's plenty of mushrooms that look like turkey tail. So just for a quick tip, it's got to have a velvety top, this turkey tail like color or color patternation. There's like brown ones, purple ones, blue ones, kind of even almost reddish colors in there sometimes. But it also has to have a polypore white bottom. So there's plenty with little purple teeth. There's plenty that have a split gill or other kinds of bottoms, and they're not velvety on top. Turkey tail is velvety, looks like a turkey's tail, and has a white polypore kind of bottom. Anyway, this mushroom, along with being immunomodulating and having all those branched polysaccharides that are amazing, has significant research around working with cancer and chemotherapy and anti-mutagenic properties. So it's really beneficial for helping cells not mutate to such a large degree. So it's got something called PSK, which is polysaccharide crestine, and it's got another polysaccharide, I forget the other one, but there's a couple in that family of polysaccharides that have shown to be really anti-mutagenic to the cells, increasing, at least from some of the studies, up to 400 times better recovery rate from chemotherapy. So this is used all through Asia as a standard protocol for anyone going through cancer treatment. In fact, since 1984, this was in the Japanese Pharmacopoeia as the mushroom to work with in going through these kinds of treatments. So we're a little behind the times here in North America, but my recommendation is do your research. Check this out. There is so much information on this. You can check it out on PubMed, or you can go into Google and type in the word scholar, and it'll pull up Google Scholar, and you get other research papers you can start to look at from there. That way you're not just looking up where to buy turkey tail, because you already know where to get it. Get it from Harmonic Arts. Anyway, the other thing about this mushroom is it has really protective properties for the liver, and also antiviral properties. So this is one of those ones that's been used heavily for hepatitis, but also was used for HIV pretty profoundly, and has a really good liver detoxifying, liver regenerative property. I wanted to mention one thing, since it just crossed my mind, because Jason was talking about mushroom toxicity, and I want to give you guys one really key thing. The best thing you can do for any liver toxicity around mushrooms is milk thistle seeds ground up like oatmeal. Put a bit of hot water in it, eat a pound of milk thistle seed. This is far better than anything they found, at least from research around what they do in hospitals. Milk thistle seed at a huge volume is great for mushroom poisoning. But only have like a pound of milk thistle seed around if you know you're going to go out and be bold. But you know, there's a saying, there are bold mushroom foragers and there are old mushroom foragers, but there are no bold and old mushroom foragers. So be careful. Okay, these polypores are really easy to work with. I think that's a fun joke for me anyway. I hope to be an old and maybe not as bold mushroom forager in my long life. So I want to talk about this one. There's actually a couple of cordyceps species around. We're not so sure about the ones here, but this is a really cool mushroom. The only reason I want to mention cordyceps is because there's something really interesting that I learned about cordyceps since this slide. First off, this mushroom eats bugs, and I've got another really cool picture. Here's some bugs being eaten by cordyceps. It eats ants, it eats beetles, it eats spiders, it eats all kinds of crazy insects. And what's really neat about this mushroom is that we're learning so much about mycology that we had no idea about. This is all groundbreaking stuff. And when I was at this mushroom conference this last year, a couple of the big papers were on the bio layer around the cordyceps. And as they researched this mushroom more, they're seeing that this mushroom has a whole host of like its own ecosystem growing around it. And they're finding that it's not just one species of cordyceps, there are hundreds of species forming a biofilm, just like we have bacteria and fungi and stuff on our skin and in our microbiome. This mushroom shows evidence of a lot of different biomes around it, or different fungi growing on it and in it. What's cool about this is that they're now being able to extract out really interesting chemistry from the cordyceps based on how they grow it and what they grow it on, and all this really neat stuff. Like they can grow out ganodermic acids, which are in reishi, that are in cordyceps. They can grow that out. They can grow ginsengicides from ginseng out of cordyceps now. So and the reason I'm only telling you about this because I think from the sci-fi perspective, in the future, potentially, you could send in your saliva, your DNA, your whatever biohack you want to put in, and they can grow out as cordyceps to help enhance your health. We'll see if that's the case, but a lot of science is being put towards this mushroom right now in the international markets to really understand, and we're talking like hundreds of millions of dollars a year are being chucked at this mushroom from Asia and Europe right now to understand it better, because it's such a cool thing. And we're learning a lot from the South American varieties of cordyceps. There are hundreds of varieties of this mushroom. What's really cool about it that we know, at least from the sinensis and the militaris, which are two of the main ones, is that it opens up the bronchioles, gets better oxygenation to the cells, and really gets the blood moving. It's also an aphrodisiac because it gets the blood moving, if you know what I'm saying. And it's really been shown to be beneficial for cutting down on things like cholesterol and producing healthy cells, working with tumor systems. Okay, I think I wanted to talk about reishi, but really...
Is it the last one, Yarrow?
I think I'm done. This is a great...
I'm like, pick the last one.
Okay. Let's talk about this one, lion's mane, for a second.
What's your big closer?
Well, I guess my big closer, I'm going to just say, is that mushrooms are something that, especially when it comes to the polypores, when it comes to the medicinal mushrooms, we are only at the edge of our understanding of how they work in the body. And there's so much more to learn about this. But what we know so far is that these mushrooms, these tree-loving mushrooms that grow in our environment, have some of the most profound effects on our physiology. And we may find that by aligning with these mushrooms, we can protect ourselves through any kind of imbalance that happens on this planet. So aligning with our forests, aligning with our forest mushrooms is the first step in creating health sovereignty. All right. Thank you for having me.
Thank you, Yarrow. Thank you, thank you, thank you. I'm going to keep us just going, but I really want to encourage folks to join in for Yarrow's webinar tomorrow night from 7 to 8 p.m. And you do a Q&A function and all of that as well as part of what you're doing. Amazing.
Yeah.
So that's awesome. So please, you all have links to that tomorrow night. And for those of you that managed to sneak in for one of his walking tours, enjoy that. And thank you always, Yarrow, for your generosity. I really have of spirit and knowledge and information. Thank you.
Woo! May the forest be with you, and all those mushroom species that Andy was talking about today.
Nice. All right, we got Andy up first thing this morning for the first activity of the day. We're keeping you to almost to the end. We'll try and keep you up for Psych Lounge too. But I'm going to introduce our final presenter for tonight. I am super excited about this this closer. Andy McKinnon was one of the first people who was brought on board for this event. We were going to do a Fungus Fest. We planned it as of last January before everything in the world changed and we realized we couldn't get together. But we are able to do it this way. And when we transitioned from our totally live event to an online event, Andy came with us, and I'm so grateful for that. He's been on board right from the beginning. He's a forest ecologist from Down Island in Machosen, and his recent research interests include the ecology of BC's ectomycorrhizal fungal species. He was a lead BC scientist for Environment Canada's ranking of our province's threatened and endangered fungal species. He's the co-author of 6 best-selling books about plants in Western North America. He's got quite the CV. If you don't know who he is already, you're going to delight with his presentation. I thank him so much for hanging in to the end and for being such wonderful company all weekend. You've been the smiling companion through the last 2 days, and it's been very, very delightful, Andy. So I'll pass it over to you. And was Don said, yeah, well, he can announce. He's also the Green Party candidate in his area. So thank you also for stepping up to serve. That's amazing. And you're also a municipal councillor. So that's beautiful too. You've got the science and that community and that service piece, which I sure have a huge amount of respect for. So over to you.
Well, and a happy science pub to everybody. Okay, how's that looking?
Amazing.
Phenomenal. That's great.
All right. All right. My apologies. So I'm going to talk about a natural and cultural history of magic mushrooms in British Columbia. This is in part, this is a presentation put together by, oh, now it's not going to move for me. Why is that? Okay, I know I'm screen sharing. There we go. So this is a presentation that was put together by Paul Kruger and myself. Can you see that?
Yes.
Excellent. And if you'd like more information on magic mushrooms in British Columbia, we've got a nice reference there to a Vancouver Mycological Society website URL with more information that's written by Paul. And we have a nice chainsaw carving of a mushroom from Masset, British Columbia. Paul and I look like that today. Everybody looks best, I think, with a cortinarius tucked behind their ear, like me on the left there, but we haven't always looked like that. Some of you may know Paul Kruger. This is what we looked like in 1980. So in the West Kootenays, when Paul and I took a few weeks to travel across southern British Columbia, collecting fungi along the way and meeting with mycologists, one of our early tours of British Columbia. So we've been friends for 4 decades now, I think. For background, British Columbia is a very large area. I gave this presentation at Telluride and compared British Columbia, for example, with California, Oregon, and Washington. BC is a little bit bigger than California, Washington, and Oregon all put together, with about 5 million people versus 52 million in those states. So we have a very large area with not an awful lot of people in it. It's an ecologically diverse area. It's a physiologically diverse area. These are the major mountain ranges in Western North America, resulting in a greater diversity of ecosystem types in British Columbia than in any other Canadian province. Different physiography, different climates, different sorts of ecosystems, and ecological diversity usually results in species diversity. We have records for more than 3,420 macrofungus species in our province. Macrofungus being a fungus that you can see with your naked eye, generally more than a centimeter across. And of these macrofungal species, a small number of them are hallucinogenic, and they're the ones that I'm going to talk about tonight. So this talk is titled A Natural History and a Cultural History, and I'm going to start with natural history and look at our magic mushrooms in British Columbia. Psilocybe azurescens originates around Astoria, Oregon. It's an introduced species in BC. Around Astoria, it grows on driftwood among beach grasses in the sand dunes near the mouth of the Columbia River. It's easily cultivated in outdoor chip beds, has been in the Lower Mainland and southern Vancouver Island and the West Kootenays, and it's introduced and naturalized in BC's Lower Mainland, a very potent species and a very beautiful species. Psilocybe baeocystis. It was very common in the 1980s on residential landscaping around Vancouver on conifer bark mulch. Today, it's still common in areas that represent kind of the interface between development and forests and agricultural lands. We find it on southern Vancouver Island as well, but it's much more common in the Lower Mainland. And as development has moved up the Fraser Valley, so has Psilocybe baeocystis. Very common mushroom that Jason referred to in his presentation around southern Vancouver Island, also in the Lower Mainland, wavy caps or Psilocybe cyanescens. It's common where wood chips, in this case, especially hardwood wood chips and especially alder have been incorporated into mulches. And we've also observed it, and I've certainly observed it, in forming stable populations in kind of disused clearings, grown in with alder trees, broom, and rubus species. It's very potent. And Kevin Trim in the Souk area has some very funny photos of this species in snow where trucks run over them, and you actually get blue stained snow. Again, a very handsome and distinctive species with the wavy caps from the common name, a beautiful caramel color to the caps usually. A native species, Psilocybe cyanofibrillosa. It's commonly found in the tangled roots of shrubs and trees in the flood zones of wild mountain rivers, but you also find it in wood chip mulches in landscaped areas. Here it is in its native habitat along a wild mountain river. A European species that's been introduced and occasionally found in grassy fields in the lower mainland, Psilocybe fimitaria. It looks a little bit like Psilocybe stuntzii in that it has a ring on it. Most of the magic mushrooms don't. This is quite a common LBM, or little brown mushroom, in conifer debris in second growth forests. And it resembles other LBMs of the forests, and has been confused with species of Galerina, which are quite poisonous, as Jason described. It's a fairly low potency mushroom. Here's a beautiful photo of it under snowmelt, but a very handsome mushroom. Probably British Columbia's most famous magic mushroom, the Liberty Cap, Psilocybe semilanceata. Some people's nominee for BC's provincial mushroom. Probably won't fly with the government, I would imagine. It grows in a very specific habitat in British Columbia. It seems to grow from dead grass roots and is especially common in wet pastures where the dead roots form a thick thatch. It's in grazed fields very commonly, but not on dung. And as development has replaced agricultural fields with housing developments and malls, and the agricultural fields that remain have been improved, that is, made better drained, the habitat for the Liberty Caps has decreased. Here's a photo from Haida Gwaii by Chris Ashurst of Liberty Caps when the events surrounding magic mushroom pickers influx into British Columbia occurred during the 1970s and 1980s. Haida Gwaii was one of the places, especially near Masset, where there were all kinds of problems with pickers invading the islands and locals. Some funny stories from the time. And Psilocybe stuntzii, named after Daniel Stuntz, mycologist, one of the few magic mushrooms with a ring on it. It's a common weed species of urban and suburban areas where lawns and mulch contain woody materials. Fairly low potency, and because of the rings, is fairly easily confused with some of the Galerina species. There are a few mushrooms not in the genus Psilocybe that contain the active principles of psilocybin. One of them is Canocybe cyanopus. They are tiny, delicate lawn mushrooms of the wet summer and the warm early autumn. And some of the related Canocybe species are poisonous. And the very handsome Gymnopolis aeruginosus, which may or may not be the same as Gymnopolis luteifolius. This is one that has disappeared a number of times from the specimen tables at the mushroom shows, both for the South Vancouver Island Mycological Society and Vancouver Mycological Society. They're brightly colored bitter mushrooms that grow on wood, and they have a very beautiful spore print. And Paneolus subbalteatus. It grows in richly manured soils, old manure and straw around stables very commonly. The haymaker's mushroom, Paneolus phenisecii, listed in some sources as containing hallucinogens, doesn't seem to in British Columbia, at least not in any appreciable amounts, and may not anywhere. In Mesoamerica, there's a long history of magic mushroom use. In this case, this is the Codex Maglia Becciano, which is an Aztec codex from the mid 16th century. You can see the mushrooms, someone eating the mushrooms, and communing with the gods by eating the mushrooms. Lots of references, at least those that weren't destroyed by the Spanish invaders. This is from the Codex Vindobonensis Mexicanus, which represents 14th century Mixtec writing, and you can see lots of mushrooms, especially on the left-hand side of the panel. However, all of these references were suppressed and not widely known until a 1957 Life magazine article entitled "Seeking the Magic Mushroom" by R. Gordon Wasson, which described his experience taking the psilocybin mushrooms in ritual fashion in 1955 during a Mazatec ritual in Oaxaca, Mexico. And to my knowledge, this article also represented the first time that these mushrooms were referred to as magic mushrooms in the title of the article. Just a brief aside that Paul wanted to be sure and include. This is Timothy Leary. He was instrumental in popularizing and spreading the use of hallucinogens and sparking the psychedelic revolution. Most famous for advancing the case of lysergic diethylamide, or LSD, but he first tripped by eating mushrooms in Cuernavaca, Mexico in 1960. The mushrooms started it off, but for Timothy at least, the LSD took over. It was in the 1960s that some folks began to realize that these magic mushrooms weren't confined to Mexico, but could be found many other places in North America and elsewhere. So let's go to UBC in 1965. That's what we're looking at here, the University of British Columbia. Paul and I tried to tease this story out. It's a bit difficult to tease out, but some students collected and ate Liberty Cap mushrooms from the UBC endowment lands. 1967 reference to this 1965 event says that the students ate them, quote, for psychotomimetic effects. That is, to induce an effect that's similar to psychosis. I think most likely they were eating them simply for the hallucinogenic effects. The RCMP were eventually involved, and this represents the first record that Paul and I know of the recorded use and subsequent police intervention of these mushrooms in North America outside of Mexico. So 1965 on the UBC campus, a BC connection. The magic mushrooms were known to exist around UBC in 1964, at least. UBC mycologist Robert Bandoni sent specimens to the famous American mycologist Alexander Smith at the University of Michigan, who in the letter on the right-hand side here identified them as Liberty Caps, quote, without doubt, unquote. So in the late 1960s, early 1970s, people, some of them no doubt tired of the poor quality of street drugs available, started seeking natural highs, and that caused a lot more interest in the mushrooms. The discovery that they grew abundantly locally in nature increased the appeal and the profile, and they became well-known, popular, and widely used in our province. By the early 1970s, many areas in British Columbia, including Haida Gwaii, had become well-known for having productive Liberty Cap mushroom fields, and were attracting large numbers of pickers from across Canada. Adverse reactions and bad behaviors by some people while picking or tripping and trespassing causing property damage resulted in pressure to make psilocybin mushrooms illegal. In late 1974, psilocybin, that's the active ingredient in these magic mushrooms, the psilocybin possession was prohibited, and it was added to the list of restricted substances in Canada's Food and Drug Act. And you can see here I've listed the years with the convictions and the provinces. You can see over those years from 1974 to 1979, the number of convictions increasing, the number of convictions from BC and the percentage from BC increasing over most of that time. But you can also be assured that most of the convictions for possession, whatever province they're in, were convictions of people who had come to British Columbia and then gone back to their home province, oftentimes with the mushrooms they collected in our province. This whole time had a fairly interesting effect on popular culture in British Columbia. Here we see it reflected in a couple of covers from the Georgia Strait. For those who weren't around at the time, this was the alternative newspaper out of Vancouver with magic mushrooms on the covers from 1974 on the right-hand side and on 1975. Also reflected by the preponderance of guides to different sorts of magic mushrooms. And probably some of you have some of these guides in your collections. There were lots of news items over these periods, and all sorts of everything from fear-mongering to jailing of magic mushroom pickers, sometimes for possession, but more commonly for trespassing. And then in 1979, the BC Appeals Court ruled that possession of the mushrooms in their natural state did not constitute possession of the restricted compound per se. That is, you could have the mushrooms, but it was actually the psilocybin that was outlawed, and a Masset resident in Haida Gwaii had been charged with possession of psilocybin after several containers of mushrooms were seized from her home, but was acquitted. And a Crown appeal of this acquittal was dismissed by the appeals court, and so from 1979 to 1982, magic mushrooms in Canada were not illegal. Here's Paul Marcano in a field near the Tsawwassen Ferry Terminal. Paul, along with his brothers Ethan and Tony, who had founded the excellently named group Make All Psychedelics Legal Everywhere, with the acronym of MAPLE, just an excellent acronym, and very Canadian, by the way. The three brothers also founded the Fane of the Psilocybe Mushroom Association, a fane being a sort of sanctuary, and they imagined that the time would come when psilocybin mushrooms, when magic mushrooms would be outlawed again, and so included in their constitution item number D here to disseminate the fact that all mushrooms of the genus Psilocybe are sacraments, and that their ingestion is an aid to enlightenment and a religious practice, therefore protected by the Canadian Bill of Rights under the Freedom of Religions Clause. Of course, there were still clashes with farmers from gatherers pursuing liberty cap mushrooms. So the mushrooms were legal, but you still couldn't trespass to gather them. And here's a CBC News clip from the time. This is from 1980. Uh-oh. Hang on a sec. That's not very loud, is it? I'm going to try and get the volume up here. Okay. Let's try that again with the volume up.
So Andy, although imagining the overdub is quite entertaining, I don't think we're hearing the actual dialogue. I'm still enjoying it, but it's just marbly, unless anyone else can hear it. Are we all in the same boat? Yeah.
Yeah, nobody can hear it.
Yeah.
But it's really funny looking.
It's super entertaining.
You can see them all politely getting into the RCMP car there. I'm sorry, I'm not sure why it's not being heard.
I think you should just give us, oh, there, it's over. I was going to say you should just like give an overdub, make one up. But
Well, everybody can make their own overdubs. How about that? But it was basically about Dieter Doberstein, a farmer who was pursuing these magic mushroom pickers on his property, who had come out from Ontario, and the RCMP escorting them to the car that they went politely to and drove off. So it's a clip from 1980 showing some of the conflicts that were occurring between the magic mushroom pickers and the farmers in British Columbia. So despite the fact that it wasn't illegal anymore to pick magic mushrooms, it was still illegal to trespass. And the police in some areas used that as a way of prosecuting people who were going into fields to pick Liberty Cap mushrooms. 1979 to 1982, the years that the magic mushrooms were illegal, were also coincidentally the years that I was doing my master's at the University of British Columbia studying mushrooms. You can see the newly released Mushrooms Demystified on the back of my desk there. That was a very exciting time in terms of mushroom guides. It was a bit of a fuzzy time. That's me in the bottom right there, along with some friends. One PhD student was actually looking at the biosynthesis of the hallucinogenic compounds in magic mushrooms using the Magic Mushroom Grower's Guide to grow Psilocybe cubensis in mason jars. We managed to talk the botany department into letting us grow them in a growth chamber right in the department, so long as the RCMP were allowed to come in once a week and count all of the mushrooms fruiting in the different jars, so long as they were roughly the same number of mushrooms as they had counted the week before. There were no problems. You can perhaps imagine some of the problems that might have been involved, but so long as the RCMP were concerned, that was not a problem at all. We'll skip a few of these here. Leading up to 1982, when the Supreme Court of Canada ended the anomaly of magic mushrooms being legal, while the hallucinogen in them, psilocybin, is not. And so from 1982 until today, magic mushrooms have been illegal. Possession of magic mushrooms has been illegal in British Columbia. At the same time, prosecution by police in most parts of Canada for possession of small amounts of magic mushrooms has really fallen off, as in the same way that it has for some other illicit substances. And you don't see magic mushrooms a lot in the news these days, except for the very exciting research that's underway that Yarrow and others alluded to in treatment of things like PTSD and anxiety around end of life time. The picking of Liberty Caps in fields has declined considerably at the same time as the availability of cultured magic mushrooms, especially Psilocybe cubensis, has accelerated. And it's not that difficult if you want to buy magic mushrooms these days to find them online or to find people who are cultivating them. A lot of the Liberty Cap habitat is gone with development and changes in farming practices, as I mentioned earlier. And some of the weed Psilocybe species of urban areas in the lower mainland of around Vancouver and on southern Vancouver Island have become scarce due to changes in landscaping materials, practices, and development. Just a few examples now about how easy and really above board in some ways it is to acquire magic mushrooms these days. Here are some photos from a recent 420 celebration, an April 20th celebration. So that's a marijuana celebration, festival and markets in Vancouver. You can see some of these booths, which are just set up on the street. They're selling dried mushrooms, peyote, the dried mushrooms being Psilocybe cubensis, hash, and magic mushroom chocolates sometimes. And Paul Kruger tells me that in terms of poison control calls regarding magic mushrooms, he started to get more poison control calls from people who got food poisoning from eating products prepared containing magic mushrooms than he actually gets poison control calls from people who are tripping out badly on magic mushrooms. So the times, they are changing indeed, quite above board. This is from Cannabis Day, July the 1st. Some of you will know it as Canada Day, but they have a Cannabis Day celebration in Vancouver. You can even get magic mushroom cookbooks now. This is, I think, if I'm not mistaken, from Shannon Birch's library, recently published. I'm keen to have a look at it. It has recipes for, among other things, psilocybin-infused gummy worms. Oh boy, can't wait. And as if there was any doubt that these things have really reached the mainstream, you can now buy your magic mushroom-infused chocolates with Visa, MasterCard, or American Express. And that brings us to magic mushrooms in the present day and the final presentation of a very fun night of presentations. Thanks to everybody who's hung in this long. And back to you, Megan.
Yay! Yeah, you guys are amazing. Well, I was like, this is pretty good retention. Thank you so much, Andy. Thank you for just so much delight that you have brought to this weekend. It's just been so wonderful to have you in Cumberland, and to somehow pretend with you that nothing is different in the world or something, just for a minute. But thank you so much for being part of it, and thank you all, everyone. Jason, Dawn, Shannon, Yarrow, and Andy again. Thank you, Kara, on the back end, helping to make everything go smoothly. Thanks to all of you. If you feel like staying...